I did not say warm, it's a hot morning. So, thank you all for braving the silent coming and joining us here today. We hope that today's event is comfortable for all of you. My name is Ritika. I am the Head of Public Relations at AIC-CCMB, and it is my privilege to welcome all of you to this World Malaria Day celebration. It's a symposium. I should not be saying celebration, but it's a symposium. I work with AIC-CCMB basically in advancing innovation ecosystems, startup partnerships, and transformation opportunities. It is an honor for me personally to host this gathering, which is bringing together eminent scientists, public health leaders, industry, and policymakers here today for this event. We are calling this Accelerating India's Path to Malaria Elimination by 2030, which is of course the call from the Ministry of Health and Welfare. And the theme this year is Now We Can, Now We Must. So today we are going to talk a lot about this, and without further ado, I want to invite Dr. Mandala Brown, who is the CEO of AIC-CCMB, to give his welcome address.
Good morning. Personal behalf, uh, I invite each one of you to have taken time out to be part of our observation of this event of World Malaria Day. It's a pleasure to have you. And as you know, it's been, the look at the numbers of malaria, so part of your more personal responsibility. From the Independence Day, where it is something like 25% of the population is malaria affected. Now we have 0.08. There is a phenomenal achievement in the category of arts, where there are a lot of reaches in the public health companies. And I'm very happy to observe this day. And also, one of the reasons why we are able to put together this program is because we have one of our colleagues here, Mr. Sedar, who is so passionate about the challenge of malaria. I mean, I haven't seen any startup founder who was so passionate with the problem in the point of public health rather than giving from commercial angle. Of course, he's interested in commercial angle of making his the product from pomegranate to reach people as a phytochemical to eliminate malaria. But at the same time, the interest. I think that's a great character where it's not only your business interest, but it's also a kind of interest to make a social impact, which is fantastic. And today we have so many offers here who are good at vaccine technologies, or disease biology, or public health. It's my pleasure to have you all today, and I have a lot of students here, and I'm sure if you look at the way the approach towards malaria elimination is being done in India. I have one slide to show you. And if you, one of the calculations that people made was 1 rupee spent on malaria elimination gave back 19 rupees in terms of, which is a phenomenal return of investment, which we normally don't look at. This is one slide I have, which kind of captures the kind of initiatives the government had over a period of time, right from early 1950s onwards till now. What it tells you to look at these, the green things where the malaria went to their peak and came down to with small numbers, but it's not a smooth curve that comes down. It is something that there are peaks in between. I'm sure some of the public health experts are going to talk on why these peaks came up in between. Is it because we relaxed, or is it our focus backward? And when I was reading this report, I felt it is very important to understand what measures have worked and what measures do not work, and why we did foresee some of the problems. These are all the great lessons I'm sure The dignitaries here would have more information to talk about that. And it's my pleasure to have you again here with us. And thank you very much for taking time. I've seen a lot of students here. That's a great feeling that students want to look at public issues closely and see how they can participate. And as Ritika was mentioning about, I mean, if you have time, please visit upstairs where our incubator is there, since you have come all the way, and see what kind of facilities are there, and also what kind of support we can offer to those who have a kind of an innovative idea and want to take it forward that has a personal benefit or also social impact. In fact, that's what I look at. I want every student to look at this as a one of the career options. In India today, innovation is a responsibility. It's not an option anymore. Because for so long we depend on others and it's high time that we do our own thing. So this is my message to all of you, the young people here. And thank you very much for taking time to be here. Thank you.
So we will start with the first expert for today. Uh, the first speaker is among India's foremost experts on malaria epidemiology and field implementation. He serves as the Deputy Director, Center for Study of Complex Malaria in India. So please help me welcome Dr. Praveen Kishore Sahu on, and he's going to be talking about the last mile runs through the forest, India's elimination endgame in its tribal and high burden heartlands. Dr. Praveen.
Very good morning. It's nice to see the next blend of academics, industry, researchers, and the leaders. Uh, as introduced, I do work on malaria since last 2 years, but I would like a correction that I'm not a public health expert at that point. As a molecular biologist by training, I have entered into the field implementation science in the last 10 years. But most part of my research has been on the pathogenesis of several malaria, the molecular markers, and finding actual therapies. So, because we see a transition of malaria cases going down over the last decade, thanks to some of the implementation projects, some of the elimination drives, the National Framework for Malaria Elimination set by Government of India 2016 for a goal of 2030, we had to restore from the bench to the bedside and the field. So that would be the kind of journey that I have traveled so far in the last 20 years. But today's talk, I will be presenting some of the nuances from our work on Dhamma. This is basically that's what the title. I think I appreciate the title, which is themed by AIC, and it just fits with the narrative that India's elimination endgame in the heart to reach high burden heartlands, and how we have evaluated the effectiveness of the Dhamma malaria camp intervention. I'll be going through two parts of this talk. The first part will describe you what is Dhamma. You all know what is Dhamma probably, but for reference, I will start with that. What is the Dhamma concept? So, and the second part I will show how we have evaluated, while other agencies also have evaluated Dhamma as a model for malaria control, and how we can put the way forward, what lies ahead, and what are the bottlenecks probably So time is short, so please apologize. I advance apologize for running through some of the slides from which will be post discussion. We can do that. But to tell you an introduction of Odisha, because this is Hyderabad, and I come from Odisha, from an endemic district. I'm a native of an endemic district, having seen malaria from the close quarters, having died relatives from this disease, which is so easily treatable, but if you don't do it, then you may end up in complications. We know that the regulatory definitions for severe complications like from jaundice to dysentery to cerebral malaria. So, without going to those details, let me tell you that we come from this state, which is about 44% of Indian population, we do contribute to about 40% of the entire malaria burden. This is as per the NBDCP data. But this is a this is pointer. It's okay, I guess. Sorry. Go back. Go back, please. Yeah. Yeah. Thank you. So, even though we have open, this is so close to my eyes, so I mean, it is really not. Sorry. So, we do have a very small population of the fraction of India, but it's contributing a lot to the public health challenge, which is malaria. We do have a land area of 5% and 82% of which is rural and 39%. Because you can see a geographical topographical map here, the ecotypes are diverse. You get every ecotype in Odisha from the coastal belt. I think some of my older colleagues are here and there. They will help you tell you how beautiful this state is, but it does present a challenge for public health. High altitude mountains, hills, forests, mining, riverine plain, undulating grounds. These present favorable climate for malaria transmission. The complex vector dynamics, we have identified so far notes we can send for, I think other speakers will cover on that. Knowing basics, vector switching so far, but there are several secondary vectors probably in the future permeating vector switches. Falciparum is the major parasite here, but EBT is also not common. But recent reports of PA and PO, and the money is also very, very evident. Diagnostics is primarily microscopy and RBT, rapid diagnostic test. The treatment rate is a first line of therapy is RSO. So I don't think I should be. Important note here is the access to healthcare in hard to reach villages is challenging. And this is what one of the major factors which sets the stage for the next slide. Now, what is this hard to reach areas and how we will turn particularly this aspect to creation of a new implementation program which was to change the face of the malaria in India. So this is just a representative area that I have been there and seen from close quarters that you have impeding factors in accessing the services. But providing the basic health services is a pretty challenging in many parts of Odisha. The lesser availability of skilled health manpower is already there, so we need to. But these are mostly inherited by tribal ability vulnerable, so it's all activities. Accessing them using a regular language, everything becomes so difficult. Even the ashas, which are the basic unit or the last unit of health care provided in every village, sometimes gets challenged. So, Heartland's villages, the pockets of asymptomatic malaria, residual is over. This became a major challenge and is a routine malaria control program which is entirely running across the country. So how do we found or envision a solution to walk the last mile, which was termed as a new intervention? How it is different from others? Now we do have the malaria control program which is called NCP. So I'm messing up. So the NCP is the malaria control program which is normally available for all plain villages. So I'll use the term for plain villages or the key areas and the other areas as the hard to reach areas. So for them, so there's a mechanism here. So thanks to Dr. Khan here, this picture is important because he's the key man who conceptualized the theory of this government also put together that of malaria camp that will address all the components of the malaria intervention that is required, which will address both the attack on the parasite as well as attack on the vectors. At the same time, with the information, education, and communication activities to continue at the outset. So difference is to note here is the green dots. Actually, I'm unable to point that red dot, but then the green dot that you see here is the inaccessible village. The routine healthcare doesn't reach there, and the blue circles are the accessible village, where the NCP reaches. Now here comes the conceptualization. To address those, you need to go there. They cannot come and do that. Right? They cannot go to the PHC there, they cannot go to the CHC there. So, what is Daman now? Daman is, we call it in India, it is malaria control in inaccessible areas. Durgama means inaccessible. That's a simple word that I use. Sanskrit is also Durgama. So, in that region, how you do malaria elimination? So, that's a simple program that to reach there, to get there, you have to be there. That's the simple concept we put together. Using mass training is not a new method, it was already there, they brought together. LLI, it's not a new thing, they put together. IRS is again another new thing. So these are all in bits and pieces, they put together in one team, made a team or Daman team. This Daman team, on a particular day, will use the village, will provide all these mass training treatment, use the antigen basically treatment if they are found positive. They do the vector control by providing LLI, they will do the IRS and the community mobilization and participation using sound boxes, music, dramas, stage plays. This really changed. This absolutely really changed. So, this definition, let me just elaborate a little bit of how they did it. That the definition of the Daman village, as per Daman official guideline is, these green villages, green dots that you see, they must be satisfying one of the 10 criteria. I have listed a few, but you can see that what could be used. They could be situated in forest areas, they could be having high altitude, no mobile, no telephone, no electricity, lack of electricity supply, and presence of high risk or tribal groups or communities, including which is now listed as insurgency affected areas where we cannot reach. So the intervention started as malaria camp method. So concentration of the camps, bring everybody together to that particular area, screen them, treat them, try. So again, TTT, the triple T method was used. Again, triple T is not a new method. But now it traveled across the difficult terrain to reach there and put together a day. And that's how, because it's based on the routine malaria control program, to a earlier method called CCMB, Comprehensive Case Management System. Again, this is CCMB. So that is what is leading to Daman's evolution. And Daman was like adaptable, like based on API, they were assigned one Daman which supposedly which is seen now observed in the malaria cases. That must be that particular village will be marked as a Daman village, and now the Daman grants will have it. So the result you see here is that seed effect. The seeding effect is that multiple rounds of Daman is going to give you lesser reservoirs at the third bowl you can see. There will be one drop coming out. And we need to extend that, you guys have enough time to catch that. So that is what Daman. Now, at the same time, it is important to survive. The important to remember is the standard of care, which is the NCP, the green box, did continue as it was. So in the routine villages, it is very important that the prevention control of malaria in plain and accessible areas, either IM, IRS, and other factors would continue to operate using the rules network of nationals. So it was important because once you take care of the high intensity, you lose control over your regular region. So that they did not lose by simultaneously, they took care of both the regions, the green and the blue. Next slide, please. Now the initial outcome. So this is just the October 2021 results have been presented. So the combined or synergistic effects of Daman plus LLI and NCP wrote out wonderful projects, which was like WHO appreciated it. We put it inside it in the world malaria report that this is the new model can be translated to the other geographical malaria world, like Africa. We have about 9% of the world malaria. And Southeast Asia, India has about 80%. So this became a massively popular program. And we all know that how the success story went into the households, into the pop culture as well. Yeah, research moreover media control for global recognized. Now on the second part of the talk. Now what do we learn here? Daman is working. Daman is not new. Daman is slightly innovative. Daman is bringing out beautiful results. What are we missing here? So at 2016-17, what me and my team we started thinking is that at the same time Daman is being rolled out, let's evaluate now. Let's evaluate its scientific approach. Let's evaluate its methodology in terms of its standard of care versus standard of care, whether it is actually working. This the endpoint was calculated by this year. And we designed a phase one study of pilot 2 years. So it started in 2019, completed in 2021, just during the time of COVID as well. This is some of the epidemiological surveys that we did in this malaria campaign that we started, which is going to the villages along with the government team. You can see that the poster does tell you two things. One is the government poster, and the this side of the part is the study team. Standing there in parallel, collecting the same samples. The second part of that name is showing that, and these are the endpoints. You know, the consenting, the pre-malaria camp questionnaires, the data collection using anthropometric measures, sample collection using microwaves, and everything was in the fixed sample. They were brought to the lab and analyzed using standard methods of PCR and endpoints were also used. This design was basically a quasi-experimental cluster assigned step cluster. Important to note here is this part, the blue part. We divided this. So, to tell you that we split this in only two districts. One district was chosen as a high burden district. The second district was chosen as a low district because both were having government camps operating. But we wanted to do the two districts because this was a pilot phase. Because the second thing was following was the scale of phase for all the middle districts. That has been the next part. But important to note that we assign villages into new groups. Like RA will have new government villages. It was not pre-assigned. Now 6 villages will be given as one. So we call it RA. RA will be created. So deliberately we will create the camps in few of the villages. And RC of villages will have continued, which is already being done. Well, so our A, B, and C over a period of time. I don't know how much of that is visible, nothing visible, but it did travel the path of agriculture. You can see the gray area, which is affected by the COVID. So we had a baseline visit. We had one follow-up, two follow-up, three follow-up. So follow-up one and follow-up two were severely and moderately impacted by COVID information. We had hospitals, we had quarantine, travel restrictions. So the follow-ups were incomplete for follow-up 1 and 2, but follow-up 3 was complete once the restrictions were taken off. So this is also a reflection of how criminal programs are challenged by natural calamities or natural pandemics like that. So what we found here is, and we won't go into it in the interest of time, intent to treat analysis suggests that malaria cancer associated with lower malaria incidence. This paper is there in scientific reports. We go through that. The relative standard of care that they present a lot of difference. We brought out the malaria cases by PCR. So this was the protocol paper, this was the finding paper, both in terms of showing that the is technically, yes, it is working damage. Malaria exposure, we did also evaluate using the next panel of 21 protein here and then over to private antigens, it did show that lower proportion of antibodies in RBC. RBC is what? Is the already given more information. So it does show you that lower molecular transmission is there, indicating potential impact of the ongoing or previous malaria campaign. This is the malaria campaign. Next. Cost analysis was also done in collaboration with New University of School of Medicine and some of the experts in the field. We did find that results are comparable to cost reported in the literature ranging between $4 to $14 per person treated per round, corresponds to about 20 to 30% of the data of domestic general health expenditure in India. So this was nicely reciprocated. The scientific findings that this is financially feasible as well. So that shows you better visibility in other parts of the world. Now comes the tricky part. What was the bottleneck? We did come across some patent malaria with a large proportion. During this research prevalence study, we found that a lot of February patients are positive by PCR but not positive by RT or microscopy. So about 61% of PMHR deletions were found in the subpatent infections. And 82% of the symptomatic cases had mostly example specific deletions. This was very for us. And we do present the quickly limitation for that. And that's what we have limited some of the diversity of the PMHR protein which was sequenced in the proportion of the study samples. And we found that similar proportions of PMHR deletions are just recently described in the Horn of Africa, the Djibouti and Eritrea. Though noteworthy there, already from RMRC Bhubaneswar and Jabalpur and the Tribal Institute Jabalpur, they already had done before our study that 65 to 60% of the PMHR deletions are already there in two of the earlier stages, although the samples were very low, the sample size was low. So it does tell you that while the diagnostic escape could be at the forefront, and gene deletions and the impact of the dynamic prevention could be compromised. Next. So, article failure in the February participants, which is more concerning to me, is that it did happen in the February participants due to they were just deletions, and it could be a threat to the elimination roadmap. A parallel study was done by the RMA, the Regional Medical Research Center in AIC-CCMB. We also did a similar was using cross-sectional community surveys, and they estimated prevalence of malaria as a similar kind of design. But in addition, they also assessed the maternal and child health as well. The findings were enormous decline as we reported in the cases detected, and PCR detected does reveal that malaria and fever as well. This is again the second alarm. Due non-clear or the minor parasites coming into the picture, and the dependence on ASHA. Although the study is financially feasible, the dependence on ASHA probably in the future will be a question. Now, the key takeaways. What are the learnings? We saw that the recent malaria situation. Now, I showed you the results from the 2022. But what happened after that? In 2027, we will see. The one came here, sharp decline, about 85% decline, so which means it did rise again, and it is on the rise as well. In 2023 graph, this is the recent data ID, Odisha is still topping the list in India, where the money is still different. In 2024, Odisha reported again about 60,000 cases alone. So again, India is back on the to take on the in its highest prevalence stage. So what are we learning here? Why can we do it? Then we could reduce malaria burden by 90 to 90%, zero malaria death. However, new challenges have emerged. COVID-19 pandemic, like any other pandemic could hit us anytime. Smarter parasite probably I would say that, probably in a gradual manner, that there is a prevention or reliability from the diagnostic. Should we move away from the premature to today's target is? new parasites, PMLPO, already provided to them. Malaysia has already reported once. We don't know. I mean, monkeys are already regulated with the recent product from Odisha, the forest. Consistency in programmatic support funding other dependency. LLM recognition, something we note here. LLM does get replenished every 2 years. If delay by even 1 month or 2 months, you can multiply by and make transmission. This is the final slide, I guess. That the phase 2 trial we are now looking at by having a non-PFHRP2 RDT and molecular diagnostics using some of the land assays in other endemic districts of Odisha, which is going to be started soon. This will be like 1 plus. So we are covering 1 plus, it will be covered by alternative RDTs and molecular diagnostic methods that land on the field to detect more number of cases than normal malaria camps are detected. I would like to thank my team, which is based in the Center for Study of Complex Malaria. This is a coming from Rourkela, having the first Rourkela steel plant in the country. We have the largest oxygen in Rourkela in Hyderabad, and we are people just like elephants in our town, like in the MIT campus just 2 months back. Thank you, and I'd like to have any questions. Yes, just to mention that we are only one of the 10 centers based in the just funded by NIH, and the International Center for Excellence in Malaria Research also. And it does reflect on several key questions. Even though I have not shown many of our findings that diseases, resistance. So these centers are responsive to change. We focus on the local facilities. We have a scientific advisory board with 2 leaders globally. Direct relationship with the National Control Program, Government, and the Ministry of Health. We are managing to produce translation research in the area, implementation research that is use of result and expectations. So I'll stop here. And apologies if I run out of time, but I can't be taking this.
Thank you for that very informative and great work. My name is Aruna Pena, and I'm veterinary biologist and epidemiologist at AIC-CCMB. Given the religion mosquitoes which you found in certain areas, do you think there is a need for more rapid diagnostic methods which alternate, because that seems to be the key in detection? Is that the only region which is very specific for detection?
Yeah. Really, because we have had the AIC-CCMB and it's downstream. I think it's going to talk on that, but there is a downstream and upstream region also. We have seen that. There are 2 cases there, but the majority were focused on the which is the coding region for the gene. I think many of us would agree that there also does not serve as a major protein which is contributing to it. So, as it is easily able to sequence it. So, we don't know. But operationally or implementation, I would never, I would resort back to non-HRP or maybe higher sensitive abilities. To tell you more, on particular WHO has already been qualified 3 of the minimum abilities. I don't know the great name, but it is LDH LDH, it is HRP2 LDH, and one is only LDH. So I expect that the higher sensitivity than the other LDH based. So LDH based abilities are a quick answer. In the phase. So in fact, the phase 2 that I showed you, we have presented the R2 as HRP2 plus LDH. So what will happen is once you see that there is no bind in HRP2, but there is a bind on LDH, you directly get an impression that it is a deleted story. So that could be part of the answer. Does that?
Thank you.
Sir will be available for more questions later. So with that, thank you so much, Dr. Samu. Please sit with this. I request Dr. Madhu to kindly felicitate. Thank you, Dr. Madhu. Thank you, Dr. Madhu. It is my honor to call upon CCMB's very own Dr. Puran Singh as the next speaker. Dr. Puran Singh is a distinguished parascientist, I'm sorry, I'm not able to. Scientist and a scientist who works significantly in advanced understanding of host pathogen and interactions and infectious disease biology. I welcome you, sir. Thank you so much.
Morning, everyone. I hope you can hear me. The screen is highly accelerated from here. So anyway, first of all, Mr. Das, Balu, and all other AIC staff, thank you for giving me this opportunity to come here and then think about why we are still talking about malaria elimination. Although we made an unsuccessful attempt in 70s, and somehow that didn't work, and one of the reasons it didn't work because the resistance of the parasite, then available drugs, and then it's resistant to PDD as well. So I'm a biologist, and I will, so interestingly, there is not going to be anything which I do in the lab. Okay, so all I'm going to talk about is based on my experience as a biologist on working in malaria for years. So my result, and may not agree with you completely, but this is what I think, and of course, in that case, we are going to listen. Dr. Sahu really nicely discussed. So there the big challenge lies. That is the challenge whether people get malaria. Particularly in a country like India of size, heterogeneity, population, and it has been a challenge. But when there are success stories as well, our neighbor Sri Lanka eliminated malaria in 1960. So, which tells us that we can do it, right? So, if you plan well. So, in okay. Yeah. So, plasmodium disease was malaria, all of you know, and but most of the time when we talk about malaria, we talk about human malaria. But I think we should know that humans are not the only one which are suffering from this disease. There are others. So there are 5 species of plasmodium called malaria in humans. Dr. Sahu talked about them. That's Vivax, malaria usually. And then the 5th species, plasmodium molsci, which is a primarily parasite of malaria in monkeys, but it naturally causes malaria, and it was found about 10 15 years ago in Southeast Asia. So when we are talking about malaria elimination, we have to take into consideration that we are not only talking about Pasteur and Vivax, we are also talking about the zoonotic plasmodium species. Because if we don't eliminate them, the malaria can come to humans again. So basically, this is where the government and the WHO is talking about the One Health, where we have to control and eliminate the diseases which are spreading from the animals to humans or other way as well. So the chimpanzees get malaria as well, gorillas, monkeys, rodents, birds, in fact lizards also. In fact, the rodents malaria parasites are the ones which have been backbone of in vivo studies on human malaria, because the human malaria plasmodium species don't infect animals, although there are some animal models for plasmodium vivax, but those mucus monkeys are not available everywhere. And studies on monkeys are not as straightforward as human can do in rodent. That's why the rodent plasmodium species are very different. Next one. So as a biologist, I have to show you this slide, and you must have seen it many times. This slide I want to show because the challenges, the life cycle of the parasite, which the parasite biology poses, control the disease, to treat the disease. And one of the biggest reasons why we have not been able to control the disease for elimination. So infection begins where a mosquito injects sporozoites, okay, so we an mosquito into the exposed. In case of human malaria, it is humans, and this stage is called sporozoid, which you can see here, and then this sporozoid migrates all the way to liver, okay, and the liver it develops to other stages and finally form the neuroderm. These neuroderms cannot go back and start developing the liver. So what they do is they are released in the blood circulation where they infect the RBCs. Again develop two different stages, finally form neuroderms again, and these neuroderms infect red blood cells. Some of these neuroderms at this stage decide that they don't want to take this route rather than become gametocytes. And these gametocytes cannot develop any further in the host. That is the one-paired host. So when the mosquito, mosquito feeds on the humans or the blood, so these are transferred to the mosquito, where in the mid-gut they develop again, finally forming the sporozoites, which are injected in the humans. So now, the malaria we talk about, we are talking about this part of the parasite biology. But this is the part that was the disease. Okay, we don't know about this part and this part. In fact, our diagnosis doesn't even see this. Okay, so basically what we see is days after the infection has been done or infection has occurred, right? So this is the challenge. Yeah. And then these stages are a problem as well, because many times we don't see them, okay, and they are the ones which are transmitted. So when it was discovered that the parasite is responsible for malaria, that time the microscope-based diagnosis, what they found the these stages, and it was not transferred to the bird malaria parasites. So next slide please. So now I have to come down, was from the right answer. So the complex multi-stage life cycle is a major challenge to control and treatment, because there are too many stages to target with a single drug or vaccine. The drugs which are given for treatment, majority of them only clear the blood stage parasites. Don't work on the liver stage. Don't work on the transmission stage. That's the major. We only have one vaccine approved in low endemic and moderately endemic regions in Africa. That vaccine is also only approved for children of certain years age and pregnant women. So that vaccine is not like a vaccine which we know for smallpox and all that. Rather, it's a chemoprophylaxis treatment. Only it provides a long-term treatment and it needs further injections with time. Okay, that's a major challenge. Too many stages to identify proteins which can be used as antigens and drug targets, because the biology is complex, and because there aren't easily available systems to cultivate in the laboratory or in animals, so biology of the parasite is limited in terms of human form. Drug resistance mechanism may differ from stage to stage. The drugs which kill the blood stage, like for example, pyrimethamine, sulfadoxamine, do not kill the transmission stage, gametocytes. So even if one has cleared the parasite which causes the disease, the stage which causes the disease, the transmission stage will still be there. So if the mosquito bites, the next person is going to get malaria as well. So that's not the problem. And of course, not all human malaria parasites are cultivable, and not all stages are cultivable. Because parasite which we cultivate in the lab is the blood stage of plasmodium concern. there are very few labs which can cultivate the insect stages and liver stages. Okay, so one needs to come together to develop complete culture of the parasite so that one can study complete biology. And as I told you earlier, the models are not there to mimic pathogenesis and immune responses in the host. And that is the challenge if you want to develop a vaccine unexpectedly. So, but here is one interesting point as well, because obviously, well, you can see complexity as a problem, but one can also see the complexity of the Plasmodium biology as an opportunity to eliminate. How? One thing, there are multiple steps one can detect. One can detect liver stage parasite, blood stage parasite, transmitter stage parasite, and of course eliminate the vector. There are multiple ways one can eliminate the disease of malaria. There are multiple unique biological processes which are known in the host. For example, the depolymerizing. Depolymerizing is that happens when the parasite into the blood cell. In the blood cells, the parasite eats off hemoglobin, and hemoglobin we know carries oxygen, and the pigment that carries oxygen hemoglobin is heme. When the parasite eats off hemoglobin, the heme is released free, and the free heme is highly toxic. So what parasite does is it polymerizes directly to hemoglobin. And the drug chloroquine which you use inhibits that polymerizing step, and that process is unique to the parasite. It's not present in the host. And of course, most of our anti-malarials, purely and other antimalarials, target that state. Then the next is parasite cell division. Parasite cell division is very unique. It is not like a cell division is in the host. And there are multiple unique parasite-specific organelles which are essential for parasite migration, across cells, parasite invasion of the host cells, development inside, and many of these the proteins secreted by the organelles are associated with the virulence. And these organelles are not present in the host. Some of them are like the microbeans, proteins, and dense present another very interesting organelle parasite has the apicoplast. So apicoplast is like photosynthesis in plants, but it doesn't do the photosynthesis. For parasite, it synthesizes some lipids. So since it is not present in the host, one can use it, etc. And the biggest challenge is the hidden parasite reservoirs. Okay. So what are the hidden parasites? What we see, as I told you, we see the blood stages of the parasite in our diagnosis, all of our diagnosis. But now there are problems. One is asymptomatic infection. So what are asymptomatic infections? So the people living in that area where the mosquito infections are frequent, with age they become immune to the disease. Means they get mosquito bite, they get parasites, but they don't show the symptoms, but they carry some parasite. So they wouldn't go to any hospital for treatment. But those parasites serve as the transmission reservoir for mosquito to transmit the parasite in a different host. That's the problem. Spleen is an organ where the plasmodium vivax stages high, and our diagnosis doesn't detect that. Bone marrow is another place where plasmodium vivax stages high. In fact, some of the plasmodium adjuvant parasites, okay, to hide there, and then the interesting organ is liver, where the parasite occurs first. Okay, that's what I will talk about briefly next one. So here what happens is I showed you earlier. So the sporozoites injected by the mosquito, they develop two different stages. But in case of plasmodium vivax, plasmodium ovale, and some anti-malaria parasites, this sporozoite decides not to take this, rather sleep. And this stage is called hypnozoite. So what happens when the hypnozoite stage is present? After plasmodium vivax infection, the person may not develop any malaria, or even if it develops, so the person gets treated with anti-malarials, but the hypnozoite stays there. Okay, next. So now the hypnozoite is a dormant liver stage, and it can cause was relapsed after weeks to months of infection without any mosquito bite. So it's a challenge, and we don't have any diagnosis that can detect technology. And it poses challenges like resistance to commonly used antibiotics are not killed by most of the antibiotics we have. And it requires additional treatment with trimartin. Okay, that's a drug which is not commonly used for treatment of malaria unless we have diagnosed that the person has plasmodiabetes infection. And then, of course, plasmodiabetes is more difficult to study because we don't know anything of that. And Dr. Sahu told very nicely that plasmodiabetes is a huge problem in India, okay, which makes the hypnotic problem even more complex. Next. How much time do I have? How many time? So I have 2 minutes, right? Okay. Yeah. So I will to talk about how primarily works. So primarily we don't know how it kills the parasite exactly, but suppose if the primarily gets metabolized in the liver by cytochrome, it produces excessive oxidative stress that kills the hepatocyte but cannot kill the other stages. But there's a problem with primary treatment. Okay. The problem is primary treatment is potentially fatal in people with glucose phosphate dehydrogenase deficiency. So it's a genetic disorder prevalent in malaria endemic countries, in tribal areas where people practice different social practice. And what happens, the RBCs, which are already under oxidative stress because of the presence of hemoglobin, and if this enzyme is not there, those RBCs have very high oxidative stress. I mean, such people are treated with primarily, the RBCs light. And the treatment of primarily causes hemolytic anemia, right? And the prevalence of G6PD varies anywhere up to 27%, in average prevalence of 7.7% different tribal groups. In fact, since this G6PD deficiency has been shown to be more prevalent in endemic areas, so it also appears that this genetic disorder co-involved with the malaria because it also provides protection to malaria as a disease. I'm not saying that the RBCs with this defect cannot be infected, can be infected, but the overall symptoms of the disease are much milder, just like sickle cell anemia. And of course, when this speed is there and there's a malaria, it is recommended to check out this speed deficiency so that the person can be treated with primarily or with some other drug. This speed deficiency is there, it cannot be treated. Next. But there is new hope for plasmodiabetes malaria. There is a plasmodium cynomology model that causes malaria in monkeys, and it also produces hypnozoids. There are humanized mice, model for diabetes malaria, and we also have human liver chimeric mice to support full liver stage development. Fortunately, recently a new drug is approved, Patinopy, which is recommended as a single dose or in combination with other antimalarials for the treatment of diabetes malaria. But this drug also causes oxidative stress, but since the Patinopy which one has to take for almost 2 weeks daily, Patinopy is taken as a single dose. So the oxidative stress in RBC is not as high. Next. Of course, India aims to malaria is by 2030, and Dr. Saul told about it. I will talk about it. I'll just go to the next slide. Yeah. So, what I think how to sustain malaria elimination, even if malaria elimination is achieved, the challenge is how to sustain it. I have my views, which are the surveillance of the mosquitoes in the field before we start knowing that the people are getting malaria. There is random surveillance for by human diagnosis and all that. There is of course our program. I believe other speakers are going to tell about it next. Thank you very much. This is what I think. We can reach the goal of malaria and elimination. What we need is the biologists, the healthcare authorities, local authorities, of course, the government commitment has to come together. And then we have to work together continuously so that it can sustain. It may be achieved, but if we don't work together continuously, malaria will come again because there are countries which do not have malaria. They get something which is called imported malaria, which means the mosquitoes can come from endemic countries to the countries which never had malaria. Okay, thank you very much. Is there questions? So yes, that's a very good question as well as this is we don't have any answer. So what activates supervisors to start the infection? Okay. So some studies, not studies actually that Buddhist people say that the increased stress or immune response sometimes because of other infections or the activation of the immunology. And it could be intrinsic mechanism that parasites project to wake up and cause the disease. Clearly, we don't have any molecular mechanism expert as of now that we can tell. So this thing is what they activate. We really don't know.
So first of all, coming to Mandala, from national world malaria hচ্ছে
Cancer bearing
Yeah.
in liver.
Yeah, every carboning parasite has to go through the liver. Calcium does not produce the liver form. Because the liver form is produced by white carbohydrate and the certain malaria, not faster, fortunately.
Yeah, so how do you try to control zoonotic transmission of malaria, like species to species, especially Hyderabad?
Yeah, fantastic. This is a huge challenge. Probably we can take an example of Plasmodium monocyte. But of course, we can't treat monkeys like we do humans. But I think is one can do it by continuously monitoring infections in the people who are living close to the forest. That is the only way. So other than that, I don't know. That's the only way. We continuously monitor whether the non-site infections are coming in humans. Because non-site infection in humans is natural. And of course, something we can't. Right? So, but we are here, humans are coming as well. So we have to monitor the malaria cases in humans whether they are non-site or something. Treating them. That is the only way. I think. Sorry.
Treatment, legal status, malaria sometimes.
Yeah, so primately is the okay. The primately is the liver stage. And the speculum means also is the liver stage, including color.
Treatment is right.
Yeah, because we don't see liver stage index. When we diagnose, we see the blood stage index. Right, but when we are diagnosing, so we don't know when the liver stage is. That's a problem. And moreover, even the drugs which are given for prophylaxis, happen like people who are traveling from developed countries to endemic countries, they are given chemoprophylaxis for long duration, like penicillin and sulfadoxine. Not so much now because they are resistant to some other drugs, but that is to protect them from blood stain infection. Thank you.
Thank you, Dr. Puran. May I invite Praveen sir to kindly felicitate Dr. Puran? So with that very insightful speech, I would like to move on to the next section. It's from, of course, from Consytel. the sponsors of this evening and the ones who have initiated this entire get together. So with the innovation and entrepreneurship are critical to the disease annihilation. This is what we are working on AIC-CCMB very well, is doing this a lot, and this is what drives us. So Dr. S.K. Dash is one of our early innovators, and with Consytel Life Sciences, he has been instrumental in developing a drug, which he will talk about. I don't want to go into that. So he's going to talk about his journey into disease elimination and what he has done so far. Dr. S.K. Dash.
Thank you for coordination. We will keep moving the slides. Yeah. So you can witness the innovation part of it here. When Dr. Sahu was here, he literally struggled to deliver his talk, but because of his masterfulness, he could hold the audience in sway and convey the message. Dr. did a fantastic job of up-regulating the innovation process. We climbed down, he did his job, but with a much better approach. Now, I have no way other than to follow Dr. Puran, so I will also do that same. But I will do with the innovation, I will keep hopping on to the stage up and down, so that at least it is the impression that there is some innovation going on. But light things aside, with those fantastic discourse which Dr. Sahu set the tone, and he did warn us that Operation Sindoor abhi jari hai. So you don't think that the malaria elimination program is over. It yet has to be taken up to the last mile. He enumerated the challenges and followed by insightful understanding given by Dr. Puran who explained that how these things, unless it is done properly, you intervene at the liver stage, things are not going to be easy. So it is just that assuming by 2030 we will be on the road to elimination, may be a big question mark. So that's where we step in. And as a founder of the company named Consytel Life Sciences, I am going to very quickly explain that what intervention we have done. Next slide, please. The talk cannot start or conclude without a homage to Sir Ronald Ross, who incidentally malaria was discovered at Hyderabad. And this is the only disease where 2 Nobel Prizes have been given. So that's the importance of malaria. And now fortunately we are sitting in Hyderabad and discussing about malaria. And just a note, like COVID happened in 2020, malaria also was equally a dreaded disease. That's why the name Malaria. Now, maybe 125 years hence, I mean, that's when 1902 or maybe 97 to 2026, more than 100, around 125 years. Maybe we all will be sitting here again after 125 years. But of course, incarnated differently, who is what we don't know. But CCMB is going to be there for the next 125 years. And then we'll be again having a discourse saying that, okay, we had talked about malaria elimination, what happened into the future. Next slide, please. That's where we have come in, and I will very quickly take you through the product. This product is called Oponica. Oponica is drawn from the peel of pomegranate, not the ones which you have, the edible ones, but the wild variety which is found only in the Himalayan region and sub-Himalayan region. So we took this peel, and in fact, technically it may not be Punica granatum, but the different species of Punica, protoplasm it is. So we actually borrowed on a mix of these. that is how Oponika was born. It is an indigenous find, and we have been into 15 more years, 15 plus years of clinical use, 16,000 patients recorded and being added more and more. And this is not just pilot basis, this is real evidence based, real world, the treatment that you can see. Next slide. So the question is, how does it happen? And as you know, when we launched it in 2013, that point of time we had two things. One is FSSAI. Either you categorize it as a good product or Ayush, that is the herbal and Ayurvedic, because these are only the two forms that possible. We could not go to pharma because this is an extract and it doesn't qualify under the guidelines of pharma. So therefore, we had to make do with Ayush because regulatory clearances are a must, and that is how you build the market. Oponika is both falciparum and vivax. It acts against falciparum and vivax. Now the question is how. We have been given seasoned discourses how it acts. There are two types. One is vivax and one is falciparum. And as we know, falciparum is also called brain malaria, cerebral malaria, which is deadly. You don't, act on that, the patient is going to, there may be a mortality. But vivax, fortunately, it doesn't kill. It will debilitate you. It will cause repeated instances, but it may not kill. So the mortality rate is much, much lower. In India, we have seen from the previous two speakers, they have said that 40% of malaria is vivax around, and but 60% is falciparum. So we saw that our extracts, they have direct anti-parasitic action. So against the parasite, there is a direct anti-parasitic action. What we have seen is in the extract, there is a composition called ellagic acid and ellagic tannins. These are primarily responsible for disrupting the structure of the plasmodium, and that is how it gets killed. Now, there is also a very important thing. Dr. Puran explained that the parasite, first when mosquito bite happens, the sporozoites travel to the liver, they get multiplied there, and then it breaks to the blood stage. That is where the classical symptoms of malaria happens, rigors, chills, etc. But that means 7 days back, actually the person has been infected, and it is in the work in process. So when it hits the blood stage, the parasite gobbles up the hemoglobin in the red blood cells. It eats up the hemoglobin, and that is how free heme is left there, which is toxic to the parasite. So what it does, it changes it into thimerosal, and that is how it escapes. Now what we do, our extracts or Oponika acts is, it actually ruptures the parasite. I mean, it stops the parasite at that level. It doesn't allow the hemoglobin digestion to take place. So we were asked the question, how do you know that your product is working there? So we pointed out, look, there are two proteases, plasmapsin 2 and plasmapsin 4. So when you do a study, there are a lot of acids. I will not go to the technical part, but just functionally, when you have those two acids, you can see that your target there at plasmapsin 2 and 4 is not working, so the parasite is not able to gobble. That is the second line of defense that the product acts. The third is beta hematine formation. Again, basically, we all have to understand that the parasite survives by eating up the hemoglobin in the RBCs. That is how it survives. But we disrupt that in multiple pathways. And it is well documented. Well documented, not sitting here and just giving something. There are 5 international publications which have been put out. University of Milan They did all of these, and that is how we know that how the extract works. There is also a very interesting part of it. Cerebral malaria, it causes lot of oxidative stress, and that is why cerebral malaria is lethal. As Dr. Sahu explained, that if cerebral malaria, the stage goes to cerebral malaria, the patient may not survive. That is where this particular product works wonderful. It has very high antioxidant potential. It quenches the free radical, and that is how the sequestration of parasite and as you know, brain vessels are very fine. Human size is very fine. Now, if there is multiple parasites blocking it, like a road jam, you have a problem. And that is what results in weak mortality. But here, there is a severe down-regulation of the promoter activity, MMP9 and the NF-kappa beta pathway, which has also been well documented, and that is how the products they work. Unfortunately, in buybacks, there is not much of in vitro studies available because as Dr. Tulansi was also saying, culturing a buyback is difficult. But till such time that we don't find a proper way, we will not be able to answer. We will only be able to answer and say that look, in the field studies, we have seen that there is no relapse, there is zero relapse of buybacks. And therefore, we have seen that if you give a complete course of Oponica for the next 6 months, there is no repeat malaria, and this has been well documented. Next slide, please. Although there are some technical part of it, if you go to my previous slide, please. Can we go to the previous slide? This shows that how the Oponica acts in terms of the MLP9 and the promoter activity, but we will not go into that. The important thing to know that it has multiple pathways, and that is how it is successful. Next. Now, this is more little bit of scientific stuff, but I will just clarify this thing that if the lymphocyte architecture is conserved, that means there is no inflammation. That means why there is no inflammation? Because the parasite has not been active. That means if the parasite is not active, the drug is working. That's the that's the takeaway from this slide. And there is an inflammatory lymphocyte activation is also brought down, basically because it has wonderful antioxidant properties. Next. A very important thing. There is no emergence of clinical resistance. Both the speakers have drawn your attention that gene deletion or the clinical resistance is a distinct reality, and we have to grapple with it. But if you have given Oponica in those patients population, there has been no clinical resistance seen. Now the question is why? Because of the multiple step weight, the plasmodium by the time it mutates and it gets into that grade where it can challenge your medicine and say that look, I am now resistant to your medicine. That probably will be very, very difficult with Oponica. So that is where Oponica would score over the other anti-malarials that are there. I also like to draw your attention that even in drug-resistant cases, this drug acts. So, as you know, artesunate is getting resistant. I mean, the plasmodium is getting resistant to artesunate, and we had the wonderful support of Dr. Kuman Singh's lab and CCMB. They ran our extracts against drug-resistant parasites, and the results were wonderful. He said, look, your product is going to act very well because it is actually acting where others are failing. Like chloroquine, resistant, artesunate, resistant, artemether, resistant, to some extent partial, but this is not. So that's the product into the sunrise level which we can force. Next slide, please. A very interesting thing. we also saw chemoprophylactic action. We adopted a whole village, and what you say is, I have myself traveled to Koraput, and we have seen that these pictures are all clicked by me. These are highly Naxalite dominated areas, I am saying 20 years back. So much so, there is a whole office, panchayat office, designations are written, but no chairs are occupied because that is the dictate given by the Naxalite saying that anyone who occupies that chair will be shot. But we managed to go there, do this intervention and prove it to ourselves that yes, it has chemoprophylactic action as well. And these figures are not my generated, neither it is AI generated. It is generated by the district education officer and the district welfare officer, which they have certified and said that this is how from 411 cases, it has gone down to 5 cases after 1 year of intervention. And these 5 cases are all important cases. People are going and staying outside, working, migrant laborers, and when they came back, they brought the infection in. Next slide, please. So that's where the clinical case came for a hepatic intervention. So up till now, we have been explained by Dr. Poral saying that look, the bottleneck is actually in the liver. Whether it is Vivax, whether it is falciparum, it travels to the liver. In case of Vivax, it sort of hides in the liver through a certain mechanism. But in falciparum, it doesn't hide, but it multiplies in the liver. What you do is just hit them where it matters. Hit them at the liver, so no progression to the blood stage, so no malaria. So therefore, even in some cases, asymptomatic people, they are infected with malaria, but you don't know. Now, asymptomatic people with gene deletion, double. So in all of these cases, actually the product can work. So and how? This is Obonica 2, the new generation product, which we have done in coordination with IIT Madras and BITS Pilani Goa campus. We developed a formulation. We were funded by BIRAC, that's a body from Department of Science and Technology, Department of Biotechnology. So they funded us under Big 16 ball, and with the help of Dr. Puran Singh's lab, we ran our tests, even University of Hyderabad, where the challenge was, as he rightly said, that culturing these sporozoites, that is the most difficult thing at the liver stage. Even in India, probably 3 or 4 labs only work on that. We successfully managed, we challenged the mice model with a sporozoid and showed that if you give our drug, our extracts encapsulated, which is liver targeted, there is no invasion by the parasite. The parasite gets killed in the liver itself. And does it cause a problem? Does it cause a drama by getting killed? No, it doesn't. Because we did histopathology, studies, we saw that there is no inflammatory responses. It is there. That means this drug is working very, very well. Next slide, please. And probably the last slide. And that is where I am fortunate not to have to explain this slide because Dr. Puran Singh has already did that. And he has said that if you can manage something at the liver, it's a wonderful way to eliminate malaria. And that is what exactly we have done. We have encapsulated our extracts. We have made it hepatic targeted and oral administration, that's the concept. Mice model we have done a challenge test, validated it. Now we will be going for phase 1 trial in humans. If CDSO approves that, and for all you are speaking about chloroquine, you know about artesunate, you know about artemether. Tomorrow, the students who pass out from the SPM department or the community medicine will read about Oponica. So that's the sunrise sector which I see, that's the promise which I see. And with that, probably the last slide is we have been awarded a patent for our all of these efforts. These are all the lab work, and my colleague Mr. Prabhanjan has been instrumental in doing it, and together as a team with experts in the field, we have somehow managed to make this formulation and validate it. Thank you so much, and the floor is now open for questions.
Question?
That's a pretty interesting conversation on the opening up. I was wondering, is there any idea what the target might be? And is there any possibility of resistance coming up if you expose it to the lower concentration over a period of time?
Yes, sir. That's a question which we are hunting for the validation, but on published literature, what we have seen is because it is acting on the calcium, the MDR genes, which is documented, that means it is different from the sites where chloroquine or artesunate acts. Now, coming to the point, what is the precise level unless we do a study of a whole sequence and then do it? For this, you are the right person to help us study, and if you give the nod, probably.
I was actually asking Puran about it.
No, we have not, but that is something, sir, which we can do, and that different concentrations actually it will not cause the resistance, because that's a explanation for another talk. But with diminishing concentration, it doesn't cause the resistance, because that's a different pathway which we can. But that answer also can be validated once we do the experiments.
Sir, this Openica project might have started 20 years back, as you said, and congratulations for that. And I want to know that quinine project, quinine herbicide, 15 years back, when I was in a tribal area, we used to treat the cerebral malaria patients in that area. So how it is different from quinine, Openica is different from that quinine. Yeah.
Quinine, the path is more or less similar to chloroquine, alkaloid function. Here also, because it is extract, it is more or less similar but different targets. Where chloroquine acts, this doesn't. Where this acts, chloroquine doesn't. So that is one thing. Plus, adding to that, even people who are diagnosed with a G6PD deficient immunology, there also this was given, it did cause hemolytic attack. And that is well documented in the papers.
Thank you, sir.
But in your mouse experiments, do you do a time series, the intervention to see when it is most effective post-infection, like they do post-treatment, they 7 to see, for example, if a human being gets infected, after 7 days if the extract is given, will it still be effective versus day 1 post-infection?
Yes, and certainly because it was a human innovation of the animal trial, we challenged it with this sporozoite. Before that, we conditioned it for 4 days of giving the drug, and then on day 0, the challenge was given. But day 0 minus 4, the conditioning was done, and then it was challenged, and we did it for 14 days. So every 2-3 days, we took up the blood sample. And finally, at 14 days, the animal was sacrificed. The histopathology was done. And that is how we would see that the parasite drop over a period of time.
I mean, the initiation of the treatment.
The initiation of the treatment was 0 minus 4 on the 4th day. After that, they continued to be given. So it is not that the challenge was given, it was stopped. It was continued to be given, but we researched that there has to be a steady state in the liver before this Orozol challenge happened. That means it points to the fact that we should be conditioning the population in the endemic area by mass rollout for let us say 12 days, 10 days kind of thing with the existing population. Maybe later when we pick up, maybe it will be a 1 day or a 2 day or 3 day kind of thing. But as of now, 12 days minimum conditioning has to be done. That's nice. Dwell much on this. But what we did was, for first 12 days, then after 3 months, again for 2 days, again after 3 months for 2 days, like that. The whole year it must continue. It is not that it was given 12 days and then we forgot about it. Every 3 months, we try to pick up the tempo and see that it at least has some stability. But that could be well reasoned when we do a randomized control or a large. Yes. 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 Who was? Sorry. Okay. There is no case and control. The whole village was administered this. So for all you know, the control would be someone else where intervention has not taken place, and those were endemically, suffering with malaria. But this 400 people who also are also exposed to the same ecosystem, but they didn't have. It was only 5 days. Would be explanatory reasons, many factors. But one, case study which I wanted to show, the other case study which I did for the paucity of time, we had 3,000 enrolled students in Koraput, different schools. They were leaving halfway and going home because they were affected with malaria. But ever since we started giving them the course, first 12 days and after that every 3 months, repeat. For the entire year there was no problem. But again, we have to actually qualify well controlled, case studies. But this is maybe at best an empirical case study. In that college, in that school, whosoever, anyone was there. Because school children, anyone was there. And then you would have made a comparison. But that comparison is easy to see. We are talking 20 years back when Odisha was the #1 hotspot in malaria. In Koraput, everywhere and anywhere you could have malaria afflictions. But here it did. So I agree. So all the four reasons that this promising product requires funding and it requires support by government and the ecosystem to see that when such products are available, which is not something often that you come across, if we can successfully validate it and scale it up, it would be your answer to artisanate or artisan. Toxicity is main because this product is highly safe. Almost 2,000 kilograms per kg body weight, no toxicity. 2,000 milligrams per kg body weight, that is 2 kg, no toxicity. We have given repeat dose. Actually, toxicity is single dose also and multiple doses. We have done multiple doses and single dose we said that anyway the administration is multiple dose. This is as fast acting as artesunate. Within 72 hours, that also clears parasite, this also clears parasite. This is for chemoprophylaxis. Yes. It could be long acting. Yes. So we will be moving towards because this sort of collaborations and trying to keep it. So thank you, audience, for your
Thank you so much, Dr. Raj. It's good to see you for a second. Dr. Kurat, may I ask you to felicitate Dr. Raj, please? Thank you so much, sir. So I think all of us will agree that all of the talks have been very interesting. Somehow we are a little behind time because of course the talks are going on slightly longer than expected, but I think that is welcome to all of us. There's a slight deviation in the program because Director CCMB needs to leave. So without further ado, I would like to invite Dr. Vinay Nandapuri, Director CCMB, to talk about what CCMB has been doing so far in terms of malaria biology.
Afternoon, all of you. I came a bit late because of my other commitments. I'm glad to be here for the World Malaria Day and CCMB's contribution. You already heard. Most of it is coming from Pura. And indirectly, the second talk by Dr. Das also has CCMB's contribution, Openica. And in that context, we have all the necessary tools to look at plasmodium in the context of molecular level, genomics, to proteomics, and all the things. So, which is an important aspect if you want to figure out the mechanisms behind the resistance or mechanisms that are associated with what would be a better target and how we actually can go about figuring out which stage something is working. These are the aspects where an institute like CCMB and the kind of facilities that we have and the group that we have can actually figure this out. But keeping that aside, I'm glad to be here for today's World Malaria Day. I mean, if you just I was talking to Pura, and basically there are around 6 million people dying every year of this. And even though we are not part of the WHO list as what they call as the high burden and high impact countries, the fact is it's still a problem in India, maybe more in tribal areas, but it continues to be a problem. And this kind of a problem needs to be attacked from every front. One is obviously the vectors, which is controlling of the vector, which is carrying the parasite. And then treatment options, both vaccines as well as the treatment options that are available to us. Lucky that we have multiple treatment options, but despite that, this is something, from the time I have joined long ago National Institute of Immunology, ICT used to and continues to work on Plasmodium falciparum and malaria as a problem. And it still has groups working on malaria as a problem. And that tells you that it is not an easy thing to deal with. It's not an easy bug to work with, right? So in the context of that, this is a continuous effort by researchers to understand various aspects. And it has to, industry to develop various other remedies. So the work on vaccine has been going on. There are vaccines, but effectiveness of vaccine is a question. So something that is an ongoing effort, vaccines and therapies. And I believe that wherever possible, with all the kind of molecular tools CCMB has, and with pulling around, you will still like to be a part of such kind of efforts. And wherever possible, we can actually look into all these mechanisms. I personally do not work on it, but I understand how disease biology works and how this resistance comes as along the way. So this kind of a small gathering that we have, and when we are talking about various aspects of malaria, is important from the context of the people who are stakeholders who are involved in this. One, it brings together people, which is good for more of a better working relation and understanding somebody else's point of view, because we tend to be in our own worlds. In that context, this kind of small meetings are very good, because then you actually get to interact with the next person at a much closer quarters. And that would actually help you in developing the relations that are necessary for actually your own research problems from the point of the country, trying to figure out other aspects. With that, I'm sure you'll have much more, one more focus you, and you will have interesting informal discussion outside the conversation of talks, outside these talks, with the speakers and among yourselves to actually contribute in a meaningful way going forward. Thank you.
That's it. Could you please felicitate Director? Thank you so much. So to our next sector, it is my privilege to invite Dr. Mandala from PHI. The Public Health Foundation of India is the leading authority on public health research, and Dr. Raghupati serves as the Dean Academics at PHI. So I would like to invite.
Thank you, good afternoon. I think we work from the perspective of creating implementation for the molecular science and to the pharmacological aspect of malaria. So I take it to the other extreme spectrum of malaria, coming to understand. So I have two components attached to it. One is trying to understand the challenges that we're trying to face. The second part of talk is focused on how climate change adaptation work on the malaria after Africa. Next slide, please. So we have a massive strategic plan because my idea is to showcase you what the industry is doing from the malaria point of view. So these targets are well known that we need to achieve elimination by 2030. Much more importantly, we need to have zero cases of malaria, indigenous cases by 2027, which is next likely impossible, but we need to have moving targets for us to reach those potential benefits of glory in the malaria elimination. Next slide. So what is known is by this very good document, Malaria Program Review. This is a ministry's perspective. So there are 5 key things which I think all of the audience must appreciate. The first one is to, as many speakers have mentioned before, is to improve the surveillance, both at level of resistance to molecule and in the parasite itself. The second thing is to look at how do you integrate in the IHIP portal. You have an information portal which the local officers in the primary healthcare centers report routinely, which is called the IDSP platform. And in the IHP platform, using the One Health platform, you need to introduce much more reporting structures. So the third key thing is to look at identifying malaria activities, which the other speakers are alluded to from the field implementation perspective. The fourth key thing which the government is trying to do is to look at the Indian capacity of block and development village panchayat. And the last key thing is to look at difficult to reach targets on the forest, urban areas, maybe the last mile research gaps. Next slide, please. So what is the statement? The achievement from 2017 to 2022 has been very spectacular. We have now more than 16 districts are got API index less than 1. API index, all of you is number of cases of malaria per 1,000 population. Now, the northeast of India has got API more than 10, more than 10 to 30, but at least it's commendable from the ministry's point of view that we have cases coming down to less than 1 in almost 80% of districts of India. Next slide. So the last mile guys were rightly captured by the first speaker. In the done initiative, we understood that 90% reduction was achieved in just a 5-year time frame. So these are some ideas to look at how to reach the last mile status gap. Next slide. I also wanted to focus on strengthening surveillance. Now, this is a very important element. There are two, as I mentioned before, there are two key important things. The first thing is to look at the molecular surveillance, as the deletion of genes has been explained by previous speakers. But from the linguistic point of view, we are looking at surveillance resistance to insecticides, because LNIs are quite important targets for us to reach that. So what resistance to the insecticides pattern in the molecular pattern in the parasite behavior is also quite important. So we need to focus on surveillance. And the last important thing, next slide please, is innovation. So innovation in India has been quite spectacular. We have the I2I insecticide spray nets. So these are sprayed with palladium and the chlorine molecule. So these focus on the I2 molecule. So a lot of studies in African nations have shown the relative contribution of IT insecticide nets to reduce the malaria disease burden in those respective countries. Reaching those addresses is a critical innovation, and one initiative has shown it can be done even in the difficult to reach target population. The third thing is malaria vaccine, which other speakers will allude to, but the matrix R2R21 matrix is a very good vaccine, and the safety profile is quite good. Clinical efficacy will come with the subsequent strains and variants. So currently we're looking at around 30 to 75%. So the wide heterogeneity of malaria vaccines, and these innovations will increase in this coming decade. Next slide, please. So challenges, I mean, this was a very good report which was published in the Nature, and they give us 5 key challenges. Challenge 1 is cure is not enough. Okay, when you say it's not enough, it means even when treatment works, it doesn't stop the transmission cycle. If you look at other vector-borne diseases in the same category, cure is quite sufficient. But here, transmission cycles also have to be stopped. That's a very important thing which we need to focus on. The second key thing is diagnosis trend is falling down. When I say diagnosis is falling down, it means that even though we have the early decades, it does not reach the last mile, the difficult to reach areas. The third key challenge is mosquitoes keep on adapting. Just like climate is resilient, mosquitoes are also quite resilient, and they have novel ways to approach the insect resistance pathway. The threat of relapse is very common in malaria. Okay, other speakers have mentioned it. Relapse is a quite important thing in plasmodium vivax. So there are perpetuating cycles of relapse in vivax, which is a difficult challenge. And the final thing is the political barriers. If you have a political will, if the health systems can function well, the challenges can be overcome. Next slide, please. So I now come to the second part of the talk, which is the main idea to show you how climate change and urbanization are working in the area of malaria. These are repeated challenges which need to overcome in the coming decade. Next slide, please. So if you look at this slide, a nicely plotted line, I want the attention to be focused on the API isoprime. I mentioned API is number of cases per thousand population. So if you look at the API lines, the area of say Odisha, some parts of coastal AP, the burden is quite heavy. The red dots are not, it's a very difficult screen and you're unable to see the slide properly. But this slide shows you what is the current status. This is a beautiful publication from the north of the ICMR campus. And this document, which was produced in geospatial risk, was the first document from Indian subcontinent to look at how malaria across the geographic map and the geospatial analysis point. It's a good reference point which I want other people to have a look. Next slide, please. Okay. Some more studies have been done. The part A, the first block talks about vector competence. The B diagram talks about mosquito mortality rate. The 3 diagram talks about parasite development rate. The fourth one looks at temperature field module. So the idea behind showing all these models is that temperature is a very important determinant which looks at mosquitoes, increased prevalence, increased burden of malaria. So there is a definite relationship between rainfall, temperature, and the parasite. Next slide, please. So the effects of climate change in the WHO document of 23 and 25 have been very distinctly portrayed in 2 dimensions. One is the direct effect. One would be on more spreading the geographical limits. So temperate zones will have more prevalence of malaria. Even though areas where temperature is around 14 to 16 ambient temperature, you see cases of malaria coming to those regions also. The second line actually is reductions in transmission intensity. So the intensity is again dependent on the parasite. So temperature, rainfall, humidity, and its related varying on that. The third thing is introduction of malaria in remote areas because of climate change and the changes in transmission cycles. All of you will understand the importance of indirect effects of climate change on livelihoods, on agricultural patterns, on food insecurity issues, on our service disruptions. So there is a definite science behind showing that climate change is a very important determinant of malaria burden. Next slide, please. So this is a very good study done by using different models. Unfortunately, it doesn't capture the northeastern part of India, which would have been much more important for this to show. So this shows the humidity pattern, the temperature pattern, the rainfall pattern, and finally something called the number of bites per person per year. So the last B panel shows you that. So as you can see, the number of the red dots of areas where the burden of malaria is much more. So this is a modeling technique which was done by groups from Bal and all in the year 2025. They used two models, Vectri and the CMP model. CMP is talks of coupled model in the comparison project. So this is the vector model first panel. Second panel talks about this is the vector CMP, this is the vector minimum, this is the bias among both of them, and finally this shows the correlation between both the models. So all the models I want you to understand that rainfall patterns, temperature patterns, humidity patterns, and the number of bites per person per year is again forecasted to be going up in specific areas of India. So the modeling tells us that if you don't work on climate change, it becomes a very important issue where malaria cannot be controlled or identified only 20. Next slide, please. Okay, so in terms of challenges, the urbanization areas, there is a lot of disparity in terms of spatial heterogeneity. Second key area is resilient and clever vectors. Vectors seem to adapt, seem to, again, there is a direct varying between climate change and the vector capacity to procreate and to engage in more uninterrupted transmission between persons to persons. Social inequity is quite important because we have pocketed urban entitlement, urbanization, which is a very important determinant for malaria incidence and also malaria disease burden. The same on local health systems. When we say urbanization, it's quite important for us to understand that most of the primary healthcare centers are in rural parts of India. The urban parts of India, when you see the urban, the hospital, the urban primary healthcare centers, you will find more challenges faced in those particular systems. Malaria transmission are covered. The last one is when you don't have surveillance systems to capture these imported cases, you will again find it higher reporting rates. Regulation seems to be a far off target from the public health point of view, but as I said before, targets mean that we take a step towards achieving them. Next slide, please. So the need of the hour which the organizers asked me was to address these four issues. One was how to integrate malaria into urban health component. Second, develop early warning climate changes. Third, embed, as Dr. Others have mentioned, embed malaria in the One Health model disaster frameworks and build a interdepartmental coordination between the urban affairs, health and environmental department. So for these four key pressing issues, what are the solutions then? We are asked to close on solutions. Next slide, please. The solutions which again have come from the malaria report of 2020-25 give us 4-5 pointers. The first key pointer is leadership. So when you have the political and commitment, funds will percolate and systems can function. When I mean political will, it is not a 1 year or a, because most state systems plan on a 3-year plans, which is implementation plans for one particular year. But the funding is not consistent. If you have consistent funds for 5 years to 10 years, then the systems can adapt and modify the results and the targets can be easily achieved. The second solution which other countries have shown is to decarbonize and to make health systems more resilient to environment. So it's much more important because we cannot control the climate change. What we can control is the decision making. So some countries like the Cape Islands, in like part of South Africa, that particular area, have shown us that decision making left the local panchayat bodies. So they would make the requirement. What usually happens in India is the districts lay out the plan, they take the forecasting for the entire mandals, and then they give out the funds. But by local decision making, the local panchayat takes the decisions informs them about it, and makes materials required in the local system. The fourth key point is you need to have more knowledge of climate change and health access. You need to monitor climate, but you need to put more focus on the surveillance systems, both for viral resistance and also for the parasite development. And finally, you need to have the climate change models to inform decision making. The reason behind showing you those vector and preemptive forecasts is because these are large panels, large systems which are looked at data from various angles, from temperature, rainfall, climate change, and humidity. And they show a consistent pattern that if you don't focus on these two elements of climate change urbanization, the target for elimination becomes a nigh impossible task. Okay? I want to end on a positive note, next slide, the final slide. So, Cabo Verde Islands have shown a success story, which again reiterate the commitment was there for long-term success. It was not a 6 months or a 1 year, but a 5 year, 10 year pipeline. The second thing is most sectors collaborated, the urban affairs, the health, the climate change, all of them work together. So the inter-department sector is there and the success is much more seen on the ground. The third key important thing is targeted investments. Investments on screening, investments on diagnosis, investments on trying to reduce the transmission cycle are the need of the hour. The fourth one is you need to build the systems which can lead to a better, healthier nation. So there are some success stories in the malaria elimination, and India can also reach the target if you need to take into account the importance of social determinants of health. So urbanization is an determinant of health, your poverty levels, your overpopulation, stagnation there, in terms of say numbers trying to reach the healthcare system to make it much more accessible. So it is definitely possible for us to eliminate malaria. Thank you so much for your patience.
Any questions?
Thank you. Fantastic. Lots of information. I was wondering whether the islands where the malaria has been eliminated, so if they also combine that with other vector-borne diseases, so I think this is something all everyone needs to look at.
The point well taken. So this was not in isolation. They looked at all the possible vector-borne diseases profile, and they admitted malaria is also the tick-borne infections there. So they're taking efforts, as I likely mentioned, probably important to work on the largest screen of vector-borne diseases, say the CC fly of the mosquito, of the housefly, work on different patterns and trying to tackle them with one segmented coordinated approach to reduce the vector-borne disease prevalence. Any other questions?
Vaccines available in other countries, for example, the ones which are inactive, I think, available in endemic area, usage in India.
As of now, total African nations, some pilot studies have been coming on the pipeline. Taking the next 5 years, we'll see more rational on those lines. So most of studies are within only the African nations, so the funding was given by these kind of vaccines tend to get funds from Gates Foundation and the Gavi Alliance for vaccines. So these studies are within only one part of the continent of African subcontinent. If the vaccine efficacy was more than 80%, the WHO recommended to other countries. But because these vaccines have been shown to be good on the safety angle, but on the efficacy angle, it was between 32, effectiveness was between 30 to 35%. So therefore, we need to see the different option. And more data comes in, we will also roll out the vaccine.
They have passed the year 2024.
Yes, that is the phase 3 trial.
Thanks.
Not from scientific community, probably also. But myself and my nephew, both of them suffered from malaria transfer. And both of them got into very serious condition. Till they became serious, it was not diagnosed that it is malaria transfer. So that is a major challenge I see. Because I'm talking about 15, 16 years back, not in the recent past. So actually, I've seen so much of screening, so much of talk, but the screening of malaria is a big challenge, and dormant malaria, why we are ready to screen and identify. So these two challenges we need to address. I think rightly pointed out, but these challenges we need to look it from the lens of the health system's concept. If you screen the entire population, would become quite difficult, but to screen high-risk population, definitely we need to work on that particular. Actually, when somebody is going for general checkup also, probably we can diagnose from one test can prescribe. Because I haven't seen any malaria or any fever tests in most of these diagnostic proceedings. Available the element of rapid diagnostic kits, it is possible in near future to see that happening. So in general screening itself, let it be included. That's what I expect. Yeah, I mean, I completely understand that. Maybe
Yeah, so as you said, the government of India will introduce the active surveillance and passive surveillance. But all the fever cases and then we were doing the surveillance. And then you're talking about 15 years on. But lot of changes have come in the government of India, our SOPs. Now the RDTs have come and we are concentrating on the malaria microscopy too. So before that, it was there were no facilities as such, but now the diagnostics fever means the first they have to go for this regular. That's the strategy.
this was done.
Yeah.
In fever, they said a couple of fevers, but they did not identify malaria.
Absolutely, sir. As you said, because before that we were doing the RDT, we were introduced. Even in spite of RDT, we are concentrating on this neonatal malaria parasite. Because this is the oldest standard, as we know very well. Microscopy is the best. So we were working upon.
Probably I don't know, I'm not aware of that. But interesting discussion, possibly of time, I think that we need to get back to the final discussion. So if there are any further questions, we are able to take your questions later on. I will pass the mic.
Thank you very much, everybody. Dr. Guru, can I please ask you something? From the morning, we were talking about malaria, and I'm always thinking about mosquitoes. And I talked about that showed me that word mosquito is somewhere in November, but I just want to say that, thank you all for, for scientifically validating that, what I believe that these mosquitoes are worse than possibilities. I don't know how, but these all mosquitoes are now creating good night and malpacha and sharp like air pressure. It hurts us, but it doesn't hurt them. With that, I'm going to call upon Dr. Badri Narayan Patnaik, because I think vaccine is something that is the only thing that can protect us, if not from the mosquito, but at least from the disease. Dr. Patnaik is the medical director of Bharat Biotech, and he will talk about the various vaccines that are in development and give us a review of them.
Good afternoon, everyone. Thank you very much for this CCMB and the other Incubation Center for inviting the industry also into this discussion. There has been a fair amount of discussion in epidemiology, pathogenesis, surveillance, and new initiatives to control the disease. And there was also discussion about the extracts of fruits and plants from elimination. These efforts are all integrated efforts to control a disease or eliminate a disease. When we were medical school students, I remember, any case of fever that used to come from the southern part of Orissa, you can close your eyes and diagnose it in the Consytel Malaria for sure. So No, I mean, before I go to the topic, I would like to ask a layman question that came up as the last question. He said he's a layman. I know from there. I'm sorry if I broke the law. You cannot be include a malaria test in a general medical checkup in any part of the world unless you are living in a hyper-endemic region where it makes some sense to see that if there is a sleeping parasite or hypnozoical or something like that, or there is a subclinical infection that can be detected. It's because look at the picture that came on your slide time and again. The liver takes away 7 days, and post that the erythrocytic cycle happens when actually you get symptoms. As long as the parasite after the bite of the mosquito is getting activated and making spores, till that time you do not have any sign and symptoms. Unless you have a very fulminating type of infection, there will end up in a case of hepatitis or jaundice. There is no way of detecting. That's the disease we have to remember. So, if your cousin or son was detected really late, that's the nature of the disease and we have to accept it. No one can help it. So that's a sad part of it, but that's the nature of the disease. Okay. So I'll talk about the vaccine part. The discussion in other parts has been done. So I'm a physician by training. I'm from Bhubaneswar. I worked in the Indian Army for about 6 years. Then working in the industry post my MD program in vaccine development for the last 22 years. So all has been in vaccines, various vaccines, but very little on the vaccine of malaria. You have to remember the industry's appetite to develop a vaccine also depends on if there is a real gap in the program to support the disease. Is there a real alarming need for the population in large that other interventions are not sufficient enough to control the disease? And parasite as such are very complicated to develop. That probably malaria parasite is the only parasite for which our vaccine, the parasitic structure, the molecular structures are extremely complex and they can cheat human immune system very easily. That's the reason probably you find every drug that we use lots of that mechanisms to develop a releasing mechanism so as not to be utilized by your body's immune defense or by drug treatment or by vaccine also. Something called a vaccine pressure also exists for the most of the crowd here is a student. For a student, it is important to understand. Even if you develop a vaccine today, there's something called a vaccine pressure. So the bug will count in terms of its process regulation to find a mechanism so as to take care of the vaccine or the drugs. Then till the time that it is not overwhelmed and the vaccine is ineffective or the drug is ineffective, we continue to strive and make progress in new vaccines or new therapies. So this picture is from the internet that I pulled out yesterday night. Again, for the youngsters to have a look. You look at these pictures. In malaria, you will be surprised it's not a disease that was discovered or found in the poor part of the world in Africa or in Asia. It became a disease problem actually in Italy. The Italians suffered the most because of malaria in the past. But because of their public health awareness, size is small, population is small, resources are higher, and of all the options, you should remember they are more disciplined than us. We suffer most of the time because of our lack of discipline. The same population that is sitting here, imagine they are shifted to Belgium or Switzerland, we'll start behaving like the Swiss and the Belgian population. We keep the chocolate wrapper in the pocket, we not throw the toothpicks. But here, without thinking second time, also all the banana pits, you throw it here. So it's not a function of the place, it is our social behavior also. Because the earlier speaker spoke about the climate change, and in fact, we are susceptible for a lot of diseases that we suffer in this part of the world. So this food, there was a synchronous tree nearby, and there was a dissolved alkaloid in the water, and people they were waiting in the water, in the story goes that they were protected against malaria. And some of you would like your weekends watch, or your whiskey would remember And some of you watch cricket, you see on the Australian grounds, there is a billboard which says VP series. VP means Victoria Bitter. So people use mix bitter a little bit in vodkas to enjoy the taste of it. So bitter, and there is still the Cinderella bitter. It comes from the alkaloid of quinine. So quinine water is still consumed today as bitter water by European countries and some of the richer ones in this part of the world also. So this is the history of quinine, where people has been used since 17th century or 16th century till the 1940s. There was availability of cinchona, bark, alkaloids, and syrups to drink around the British time. Later on, it was purified in pharmaceutical excellence to keep to tablets and syrups, so it became more palatable or more easily transferable from place A to place B for control of the disease. And the paragraph for interest of everyone is the Almighty has created the humankind in a way that without a drug or vaccine also, the nature takes care of it. You imagine the places in the map of India where time and again everyone has shown the Odisha southern part and the western and northern part are endemic for malaria. But remember that part of Odisha also has the highest amount of thalassemia, sickle cell anemia, and that's nature's way of protecting you from a severe form of transparent malaria. Because people who have got such kind of hemoglobin as this are protected partially or completely against the severe form of malaria. Next slide, please. It's more like a storytelling without taking into the complexities of vaccine development. Because when we go to Africa and many parts of India today for any national program or talking to the policymakers, there are very educated people who would ask, polio trials have been done 50 years back, what kind of trial you want to do? Because I'm someone who do clinical trials for various vaccines, and trust me, there are educated people today who ask this question. In my own state, Odisha, at least 15 years back, polio trial was done 25 years back. What kind of trial you were thinking of? I went to do a meningococcal vaccine study in 2010. So this is the third part of understanding. So you guys are the next generation Z plus. So please try to educate everyone that we should not be talking like that. Huh? So look at malaria persistent threat. Most deaths today's world happen in Africa. Everyone has showed about India's story, and some question was, you asked the question, are these vaccines available in other places of the world? It can be used. He said, all vaccines are, I'll not come to vaccines. So the number of cases globally in 2023 was 250 million, and 569,000 was the number of deaths. This is a global picture. And under 5 mortality contributes about 25% of this entire number, and the species which is responsible for it is Plasmodium falciparum. So the focus of the discussion this morning is around falciparum, because when little children are small, the infection that mostly we had in Bhubaneswar or Hyderabad is virus. We used to take chloroquine, used to be fine. When we became physicians, at least I as a practice I did, I always take chloroquine 7.5 milligrams for 14 days to everyone that I treated, assuming this is the virus. And this cassette test of taking falciparum or virus, it used to exist. At least in 2097, it was already there for everyone. So it's not something that was discovered recently, it is a thing that is there since time immemorial. Sub-Saharan Africa is a place that Ganesh Mishra, my other industry colleague, and we travel most often. And these are the countries in Africa who have accepted and rolled out these vaccines. Next slide. Vaccines have been very, very effective. Apart from safe drinking water, remember vaccines are the number one intervention that has decreased or abolished human suffering today. Because if you remember, any Hajj pilgrimage to Puri, Rath Yatra, any pilgrimage to Mahakumbh that's been happening for thousands of years, people were given typhoid and cholera vaccine. And you can see them in the old British paper which have turned round today. If you have a family which has got a habit of archiving old documents, you still see written in the pen, PABTT given, severely painful vaccines. So vaccines exist in India since pre-British time also in different forms. So the malaria vaccine introduction status, if you look at Africa, because this is the only continent in the world that has rolled out a malaria vaccine program. I'll come to the vaccine later on. And just these are the slides that I have borrowed from my own department, from the advocacy data that we use for sub-Saharan Africa. Dr. Das, I'm sorry, I have not made slides for this program per se. So if you look at the areas which have rolled out vaccine program in sub-Saharan Africa, a light green, brown, and the deep green, and the deep green ones which have already implemented vaccine program in the NIP. Because in some part they say PNI, and some part they say NIP. So these are only few countries. If you look at Cote d'Ivoire, Senegal, and Benin, the countries in West Africa which have accepted them in the PNI, and there's a small Central African Republic which have introduced into the national program. Others partially rolled out in their program, or they are catch-up vaccination kind of thing. And they also depend, as you said, on Gavi funding and DFID from the United States, which is again there right now. Because all DFID people have been pulled out from the African continent as we speak today. And he and me were there in a program in Nairobi when the President of the United States declared all people of USFDA, the state DFID, to be moved to the mainland within 48 hours. They don't mind whether you are fed, you are for children's school. So that's the problem of funding today that exists for the vaccine program. These vaccines are expensive. Malaria vaccines are not like diphtheria, tetanus, and pertussis vaccines or the beta vaccines. So the estimated malaria death that happens is mostly in Niger, as well as Central African Republic. Next slide, please. That's the African story. This one I introduced and borrowed for something for this program only. So you can see always the maroon colored ones is my state of Orissa. I'm very proud of being from that place. That state, it houses the maximum amount of malaria. And I remember there was a time, as I told earlier, we treat all fevers as malaria in medical college when I was irrespective of testing. And it comes 99% time because everyone comes from either Hyderabad district or Korakut, Kalangi, and all that, and they all are transferred positive. So the malaria deaths in the vaccine introduction status is very sketchy. There is no vaccine introduced in my country. Next one. If you look at this particular slide, this talks a little about vaccinology. Transmission blocking vaccines do not exist today. And people like me, him, and him who are for a small startup company or a small biotech, we try to work on all these vaccines. And we all know who are startups or industry, it is extremely difficult to generate and sustain development of vaccine candidates over a sustained period of time from 10 to 25 years. So transmission blocking vaccines are extremely impossible to work with. There are some established companies as well as companies who have got very high amount of venture capitalist funding. They use this in the US right now, and what we in the US Army also doing it is that irradiated sporozoites that has been used to block or produce antibodies against sporozoites and try to block transmission. But it was difficult because the generation of plasmodium falciparum sporozoites was also a difficult exercise like culture of sporozoites in lab settings. The one that has got a vaccine is called pre-erythrocytic or hepatic stage, and these are two vaccines. So the concept of vaccine, at least in pre-erythrocytic stage, is to generate antibodies to interfere with parasite development in mosquito that doesn't exist. It has no impact if you make a vaccine for mosquito, it doesn't have any impact on clinical disease that exists in the population. it is part probably of the elimination strategy which can be combined with mosquito net that are impregnated with chemicals or the sprays that we use. The prevention of liver cells in the hepatic stage prevents infection, impact on the infection threshold, and impact on the clinical course of the disease and prevents transmission. In this regard, next slide. These are hundreds of vaccine candidates that all of us work on, but I focus on the ones which are real and exist today, not on the new vaccine, because this is not a vaccinology course. Otherwise, I would have gone with each one of the stage. Next one, please. Yeah. So this is the vaccine from GSK called Mosquavac or Mosquavax. These have been tech transferred to Bharat Biotech in Hyderabad. And we at Bhubaneswar have got a plan for precision biologics. And we have started the tech transfer and initial manufacturing process of this vaccine. This vaccine is pre-qualified by WHO, which means all procurement agencies like UNICEF, Gavi, and PAO can buy it and supply it to the respective countries at various levels of prices. These are decided by the GNI and the per capita income and the financial capacity of the countries. Some pay full, some pay half, some pay a small percentage, and as a function of time, you start increasing your contribution to the country, and as you become self-sufficient, Gavi goes up and you self-fund. Like India today is not a Gavi eligible country, but remember when we started pneumococcal and HPV vaccine, bulk was 50% was paid by Gavi and 50% the country, and we are not a poor country anymore, so we buy our own vaccine for our country. And in India, there are enough good drugs, and it's more accessible in most places. So the drugs that are available are enough and efficacious. So maybe the policymakers haven't thought about vaccinating this population. Because vaccine is a public health tool, then you do not do it for just one province or one taluka or a state. So maybe some reason would be there, but we would be happy as suppliers to sell the vaccine and make money. But from a public health point of view, government of India has not given any hint to us that in 2032 or 35, I want to launch another vaccine. Let's engage in a discussion. Are you interested in doing a study in my school or a country? Nothing of that sort has happened. So that means I agree that the dip in the number of cases of death have happened and is successful with drugs, with sprays and nets and improved healthcare status. So this vaccine which we use is a repeat of a T-cell epitope. So it's called repeated T-cell epitope from the southern protein, which is CSP, and these are expressed on hepatitis B surface antigen, HBsAg, to the vaccine that everyone knows, hepatitis B vaccine. So on that, it grows like mushrooms. So it's like a sunflower. I think all of you have seen a sunflower center. So the small spikes are the anti-malaria antigens, and the bulk is the hepatitis B surface antigen. We use that for as antigen along with the adjuvant called ASO1E. It's a patented antigen of GSK. They will not transfer the technology how to make the adjuvant to us. They will be both separately to us. We then later on we make just the antigen and we depend on GSK to supply ASO1E to us to make the adjuvant and make the vaccine and supply it. We are working very closely with various African Union countries as well as Gavi to ensure that we have the enough number of doses to supply to all the countries that have rolled out the vaccine as well as plan to roll out. We have a competition. In the next slide, we'll show you next one, please. So next one. Next. Yeah. So the other vaccine which is called R21, this belongs to Serum Institute of India. This was discovered in Oxford in UK, and the technology has been transferred directly to Serum Institute, which is one of the largest vaccine manufacturers in the world. So they and us are the two players as of today to supply anti-malaria vaccines to sub-Saharan Africa and other parts are necessary. Vaccines, both vaccines have got a very weird way of vaccine efficacy between 30% to 78% of vaccine. Combined with chemoprophylaxis, this vaccine has got a slightly higher efficacy compared to standalone vaccine.
So various countries use various processes. Some use impregnated nets plus vaccine. Some use pre-season chemoprophylaxis plus vaccination, depending on the availability of the funds. But the fact remains that both all these sub-Saharan African countries depend on GSK or Serum Institute for now, and in 2 years' time, it is Bharat Biotech and Serum Institute who will supply the vaccine for the entire sub-Saharan African. Well, 4 doses to be given starting from 6 months or 5 months. 6 months, I'm saying, because 246 is a PNI schedule for the diphtheria tetanus continuing vaccine, the first 3 doses. So the last one is the visit that countries use to give the first dose of malaria vaccine. So 3+1 is the schedule, 4th one is the booster. These vaccines are expensive because this took millions and millions of dollars to make, because I thought parasites are very smart ones compared to virus and bacteria. It's difficult to make a vaccine. So the next one is so Mosquitovac is the name that we have given to our vaccine. Mosquirix is a vaccine. Rix, any vaccine that makes RIX from GSK is from Rixensa. It's a place where the plant of GSK is. So it's called Rix for any GSK vaccine. So the Rix, wrote the Rix, all Rix of GSK are Rix. So ours is Mosquitovac. We will make it in Bhubaneswar. This is a safe and efficacious vaccine. We have used at least 1 million people in our clinical trials in the past, and it has been proven to be safe without any major adverse events. This same adjuvant has been used in other vaccines of GSK, and GSK, being a big multinational companies, do not want to make a €3 or $3 vaccine anymore, so they have transferred the technology to us. We generally make $1, $2 vaccine in India. So that's the story of RTSS ASO1E, that is anti-malarial vaccine. Next one. Thank you very much for your attention. If you have questions or you have to answer on this one or any other vaccine if you want. So to do a vaccine study in malaria, pregnancy, lactation, and in HIV is always a difficult proposition. We did not do a pregnancy study, but post-hoc we have seen women of childbearing age who have received the vaccine and they conceived, and we have followed hundreds and thousands of them, and it is safe. And likelihood of the babies or the infants getting malaria post-baby because of transplacental transfer of IgGs we will see, and there is fair amount of protection in the babies of the newborns.
Talking about the vaccine.
The vaccines are given in a schedule of 4 doses. The first dose is given at 10, 20. So 14 to 18 weeks is the first dose, or the 6 months is the first dose. 4 weeks interval, another 2 doses, and 6 months after is the fourth dose. 4 doses. Many countries have accepted 3 doses as their, because it all again depends on their compliance, how robust is the immune system, and the army activity.
Could you tell us why the efficacy is so low?
Okay, so I'll tell you a small story now before answering your question. 15 years back, we did a study from Sanofi in Thailand along with the water league of US and Thailand, and we were looking at prostitutes, and people have a habit of visiting prostitutes, and we found that efficacy of 18% against HIV contracting in prostitute goers or soldiers or merchant navy people who visit prostitutes normally. And 18% efficacy gave us a place in New England Journal as well as Lancet. So your appetite to accept efficacy depends on the disease. If tomorrow I send a rabies vaccine and say it has got 80% efficacy, people will beat me on that unless it is 100% efficacy. For a disease like malaria, Consytel or HIV, or tomorrow if there is something against Zika, with the efficacy of say 50% also, you may get a lower price. So it also is a perception of
the modality, the complexity of the work also decides on what kind of efficacy is acceptable, which is not. Did I answer your question?
It's partly, no, as you grow a little bigger from your student days to a junior professor that you are today, your understanding of disease pathophysiology as well as outcome in terms of public health benefit also will decide acceptance of the efficacy number. Because for rotavirus, globally, in developing world, between 50 to 60% is a fantastic number. Because the indirect effect of vaccination also decides how great the vaccine is.
I had actually two questions. One is that in case giving a primary course of vaccine doesn't confer lifelong immunity, then in that case, since it is not fatal as such, like rabies, why do we have, unless we reach a threshold level of, let us say, 80% levels or so, why do you need vaccines in the first place?
So now I'll take you to sub-Saharan Africa. In sub-Saharan Africa, who are the most important candidates to receive a vaccine, or who are the ones who suffer the most? It is the underfunding. If you look at the socio-economic behavior in Africa, the mother will go to the traditional healer or the, how do I say, the ones who wear the animal feather and all in the head, the village, how do you say, faith healers. 90% of the mothers in traditional Africa or sub-Saharan Africa go to them, and only 10% come to the village. And in the 10%, 50% are school teachers and government employees and doctors. So the mortality 20 years back was extremely high because of malaria, because of not seeking for chloroquine, cleaning, primary and artemether for adults. So the only way to increase life expectancy in Africa was to vaccinate rather than depend on the time-tested, highly efficacious drugs that we continue to use in our country. So we use those traditional healers, magicians, faith healers, school teachers to reach out to vaccine as well as the Pandit, Maulvi or people like that in Africa today also, and the pastors to reach out to people rather than the doctors, because people have more faith in traditional healers. Well, that's the way the countries are. When you go to that part of the world, you realize that all your medicine and medicine that you are looking for pediatrics and medicine doesn't exist in that. So we have to also adapt to country specific and region specific needs. Yes, please.
Hi, good afternoon. Thank you for your talk. I just had a couple of questions. During the pandemic, I think I was seeing some news regarding, I think either the WHO or the other Gates Foundation basically sponsoring a trial for malaria mRNA vaccine. Then obviously you're a vaccine expert, so this is obviously you mentioned this little while back. There is still like a little bit of an efficacy gap to overcome. So the thing is, so what kind of roles do you think, like traditional vaccines, like what Bharat Biotech and what we generally make have perfected over decades, and what's the gap, let's say, an upcoming mRNA vaccine, because I think multiple people are working on mRNA vaccine candidates in this space. I will just, and one not necessarily from this space, I just wanted to hope that you could share some more light in terms of the gaps or the efficacy gaps and the role like a next generation vaccine, which is obviously still has some way to go to be perfected, to be deployed over the long term. What kind of role can, let's say, the mRNA vaccines can play in the space of malaria specifically? Thank you.
Yeah, very nice question from a general vaccinology perspective also. I mean, when you ask the question, and many of the people who are young and who have read about mRNA vaccine or newer vaccine per se, should understand the understanding that today we have on mRNA vaccine is that there are two things which are extremely important. One is the LNP, which is a lipid nanoparticle, and the second one is painted around the capped or the uncapped mRNA that we use as a template, so as to fuse into the formulation so that when you inject, the template also gets integrated into the cell membrane and goes inside the cell machinery so as to mimic RNA. mRNA is messenger RNA that goes into the cytoplasm and hoodwinks the cell that this is one of your own RNA and start making the protein that you desire, right? So this is the overall game of it. I was in Cambridge that time when the pandemic happened and we were working on the mRNA for COVID. We tried our very best working day in and day out to make a vaccine, mRNA vaccine which is stable at -20. Trust me to tell everyone, we failed miserably and we could not go below -50. I mean above -50. The vaccine was -80, then we worked on -70, then -50. Any temperature that was increased to -10, 20, denatured the mRNA and we could not make a vaccine. We heard in US that time there was a company in India that made a 2 to 8 stable mRNA vaccine. And later on we realized I do not want to comment on it because this is a proprietary information of that company. This was an NIH research group, and for some reason, in a way, this technology was brought into India by some mechanism. And there's a case that is ongoing in the Manhattan court today against the Indian company. And whether it is closed or it is still ongoing, I do not know. So for a country like Africa, a continent like Africa, for a continent like Asia, where today also we struggle to have a INR, ice line refrigerator for oral polio vaccine or rotavirus vaccine, to have a -80 infrastructure to make a vaccine and make it available will be a big task. Even if you manage to make it, the cost of production of that vaccine will be the tune of $15 to $25 per dose, where in India and Africa, the policymakers negotiate with us for a 15-cent vaccine also. Forget about $1. In last month, we were in Nairobi and the National Technically Advisory Group on Immunization of Kenya was asking us for a 20-cent typhoid conjugate vaccine. Your cost of good is 100% 5 cents. But in order to sell a vaccine of 20 cents, you are missing out on the link, a missing link like Archaeopteryx between the production house in Turkapalli and Chametri to Nairobi. There is an air freight, there is a travel of Bhakti Patnaik and Mishra to go to Nairobi in business class, live in a nice Hyatt and spend money. Where this money will come from? See, it's normal for policymakers to ask, my children are dying on the street. Thousands of children are dying. You are interested in making money? Imagine how will you sustain the R&D? How will I give you the salary? To be optimistic, to have a $1.52 is a good way to go than dropping slowly down to 10 cents and 5 cents for a vaccine. So let's not use a vaccine. You decide that I will use chloroquine. I will use the fantastic discovery that you have done of Unica. Why not? If a country doesn't have money to afford a vaccine, they should use this traditional mechanism. Say that I use 10% of vaccine or 90% of that fantastic drug which is there from the South Himalayan pomegranate. Fantastic. So, informed decision has to be made. You cannot expect to get a vaccine with 80% efficacy and willing to pay 10 cents. I'm sorry, you cannot. I might sound very rude, but this is the reality. When you all become entrepreneur tomorrow, I as a policymaker in Government of India will ask you, give me the vaccine in 10 paisa. My children are dying on the street. This is a dilemma. So mRNA vaccine to make is supremely easy. Any postdoc can make you mRNA vaccine, but trust me, it is not viable in multiple parts of the world. Israel can do it, United States can do it, France can do it, but it has its own downsides. I can tell you example of some of my friends, some Italians, some Canadians. In my floor of 80 people in second floor, out of the 80, 79 who have received mRNA vaccine from Pfizer, Moderna, BioNTech, we have bilateral avascular necrosis of the fever, and till today they have titanium heads in their fever. Whether it is because of COVID per se,
mRNA vaccine per se, it's a kind that will answer. I'm not the expert on respiratory association, but this is you have to absorb. You do that in India, and 3 children land up because of vaccine, and someone says, an intelligent person like you would say, I think it's mRNA vaccine. You will see the doctors and the company will be waiting up to death on the streets. So we all have to live with the level of education, the amount of money, the infrastructure that we have to store the vaccine. It's a holistic approach that a standalone cherry picking of a vaccine can be. Thank you. Any more questions? Write to me, I will answer your, I'll leave my name and I am happy to answer your questions.
Just a minute, Dr. Chakraborty, to do the honor of the So we move on to the last section of this event today, and we've got a lot of great lectures and we've got a lot of new things that are coming up. So it is my distinct pleasure to announce the next panel, which is the Now We Must and How We Are Going. So, Dr. Anuradha, she is a Senior Regional Director, Ministry of Health and Welfare, Government of India. She has she will be the moderator for the session. Dr. Madhu has given this position to Dr. Puran Singh because he feels that the discussion should be more on malaria and anything else. So I would like to invite. Yeah, sorry. Unfortunately, we could not get the auditorium, so we have to make you. So, Dr. Hamada, I'd like to invite you on stage. Dr. Koteshwar, Dr. Badri Narayan Patnaik, Dr. Kishore Sahu, and Dr. Puran Singh. Could you please join us on stage?
Good afternoon to all. This morning, it's a great discussions. We are listening a lot of things about the malaria. First, I would like to thank my CCMB director and all the participants who are here, and then Dr. Das and everyone who is here. Today, first, I would like to congratulate the CCMB, which we are celebrating World Malaria Day. It's the first time we are celebrating it in Sri Lanka. Before that, just we think about the younger generation. Okay? You are the one how to take the malaria elimination by 2030. We are not talking about. But think deeply. How can we do it? So this is the brainstorming for everyone in the government of India. And not only that, but not in the public sector, in the private sector too. Everywhere the country has to go up means there is a collaboration between the public and private sector. And the government of India is working on that. And on this line, just I would like to say how we are, government of India, we are focusing on World Malaria Day. And Sir Dr. Raghupathi has told the many of the things, what are the challenges and how we are going to the eradication of malaria by 2027 and 2030 without the indigenous case should be there. We have four pillars, as you know very well. We have a prevention, early diagnosis, early treatment. There should not be a proper reference should be there. So the first point as a prevention, we will focus on the prevention. Madam, as you know very well, the prevention is better than the cure, as you know very well. The state governments, with the collaboration of central and state governments, we have a lot of projects under the surveillance and the diagnosis and the treatment part. As you are in the community medicine, the medical colleges and the director of public health, okay, how you can guide the medical students or the PhD students in the surveillance part, okay, what are the changes you need by the policy makers to do it in the surveillance system?
Thank you, madam, for asking the nice question. And actually, I'm not a director of public health. I'm a professor and actually in the community medicine department. Actually, I'm not dealing with the direct, what to say, health side. We are in the academic. I am an academician, and for PGs and UGs we are teaching. And now government has introduced family adoption program. So in this family adoption program, we are taking the students and making them to adapt 3 houses per student. We are having 250 students, and these students are adapting the families. So in these families, if anything happens regarding malaria, now I can after this very good session, whatever today's session, I can introduce this malaria also in the family adoption program and in the surveillance part. I want to introduce this malaria surveillance also, and we try to prevent related how the disease is causing, what are the physical dynamics, all these things we came to know, and available options for prevention and to prevent the transmission also, I came to know now. So that's why I came now to learn only, not to teach everybody. So I will introduce, I will teach now my students completely. Thank you.
Yeah, thank you, ma'am. Because this is the thing and then which we were expecting. When the student in the medical college, with the PhD when they are doing, that time only we should know about the prevention. And then we should know about our goals. It may be TB, maybe leprosy, maybe malaria. When we are aware of those things to children, even in the research, they can do a lot. Thank you, ma'am. Yeah. Just, I would like to, this is for Dr. Madhusudan Rao. Are we talking about the prevention, about the bed nets? Okay. This is for you, sir. As you know very well, and then we have introduced a prevention alliance we have introduced. We have reduced the death, malaria deaths, and as well as transmission also we have reduced. Do you think whatever we are, as a traditionally we are using the pipe drawing, there is a resistance also which happened. Can you focus on the new interceptor on GP? Next.
Probably Madhu could have answered this better, but anyway, now I'm so now I'm feeling that, but I think it's good that it came to me. Although I didn't work on that part of the malaria research, but I'm kind of aware of it. So the one thing is I think what many people are saying, surveillance and surveillance. So insecticide treated badness. They have worked not only in India, in Africa, significantly, because they are the first barrier which separate potential patient from the mosquito, right? And Generally, the mosquitoes which transmit plasmodium are the nocturnal. Right? And what has happened in Africa, we have seen that the mosquitoes develop resistance to the insecticide which was being used to lace the net. So what we need to do is timely surveillance whether the mosquitoes have become resistant to the insecticide. If that is seen, even in two cases, so change our program by using different insecticides. So, and I think that is what is needed. And probably that should be now told to the companies which are making this in nets. Not maybe just to the people who are involved in lacing the nets with the insecticides. Okay. I have an interesting story to tell you that even if a mosquito or a parasite becomes drug resistant, and if you remove that drug or insecticide from the field for certain amount of the time, the drug resistance is lost. And there's a very interesting story the research people have done in many African countries. All we need is timely monitoring of the resistance to the insecticide. Okay, and replacing the insecticide with the one which is still effective against the mosquito. And with time, I'm sure the mosquitoes which became resistant, they disappear. Okay, and the white man comes back. That's right.
Do we have any research on this? Because we have a resistance on this, meds and all. As you know very well, there's a new substitute meds also, which are used in African side. So, do you think that we need in India also to develop such meds?
I don't know on top of my head right now, but there are books in India. I think he is a better person to answer this.
Yes.
Thank you. I'm not an entomologist, but working on Daman has produced this human parasite and entomology angle. So probably from that perspective, the resistance that is getting researched on the insect side, India has molecular resistance at the level of insecticide, so it's not well characterized. My team is also working on another corporate committee for the next NIM or this is my team, Dr. Hart, who has done wonderful work on entomology. We do a screen for the WHO methods of insecticide resistance, the susceptibility test, synthetic for DDT, which is banned, but still we use as stability, and tetramethamine. These are the three drugs. Finally, either you find them in the IRS, the insecticidal or residual spray, and or in the aerosols, or IT, which in the African setup you can call them IT. But in all, very you find all these three chemicals prevalent. But then the molecular panel that is being used in some of these as was already discussed in African setup, there is a satellite, microsatellite resistance, the panel that you see, and then the VNTR, the very number time to piece, and there are some of the assemblies. These things are largely magically used. I don't find any so-called new papers from the Indian context to characterize these insecticide resistance in our vectors. Though, having said that, on the parasite and the human animal, you can see that the resistance probably at the drug and the diagnostic. So, I think the resistance at the diagnostic, like the commercial particle we might talk, diagnostic resistance, which is the RDT, failing, if I can say that, or the drug reaction, which is again anti-factor of an office conduct, but not yet enter, I would say, as per of this report, although sporadic reports will contradict to that. So probably the resistance at the insecticide, the resistance at the drug, and the resistance at the diagnostics. Probably will be the biggest challenge.
In fact, and in infectious disease per se, I think similarly is not a very lucrative managed for many of the Swedish passing out from whether it's a university, there has to be a government mechanism, either in the or in graduate schools or postgraduate schools, to there has to be certain level of what we call encouragement for sending abroad, sending to research institutes to come out as good entomologists. Because this seems like, in my experience, a very not so lucrative branch.
Which is very, very important. We are living on one health. We are talking about one health. So technology needs to be strengthened because as our students also they don't know much about technology. We are talking about the insects, we are talking about everything, but there is an integration, something there is a mechanism, as you said.
I can tell you definitely, 20 years back was not so lucrative branch. Today, because of private practice, because of adoption, as well as other things, a lot of veterinary papers and lot of common conferences that we attend along with the veterinary things. So maybe this is time for you as policymakers to bring in the entomology as a lucrative branch with a lot of incentives so that that department is also taken.
Absolutely, sir. As you know very well, so now every district we have a technologist. This is the model of the China. They have a very good system in the mosquito control. All the metabolic disease control because they have a state metabologists, district metabologists, they have a mapping of high risk areas. So it has to be taken to the other states also. And a few of the states it's not there, even Orissa also not there, even Northeast also it was not there. There's just one or two metabologists that are driving the things.
So even in the department, in man's department, if you go,
Yes.
you will see nice pictures in the class covers, you will see this side, this is Egyptian, this is the US Africans. But nothing beyond that transfers young students like this. You ask how many of them want to be an entomologist? You will not see any hands being raised. Because that's not so lucrative or not well funded.
Yes, we do have post of entomologists, but they never fill it. And even field also, they not trained in the respective subject. Just promotion or to they want to stay in the Hyderabad, they will come. That's it. They will not teach. They will
If you
not.
look at a small example now, say 20 years back, no one wanted to be a virologist. Everyone was looking at bacteriology because you can see a bacteria. Now, if you ask, we struggle to hire bacteriologists because everyone wants to be a virologist. Particularly after COVID,
Yes, yes.
no one wants to be bacteriologist. This is very boring. So there has to be a way how to make these people more interested to become an entomologist.
10 seconds. Somehow you find well-trained entomologists in agriculture industry. So they focus on pests. And most of the writing stay focused on the plant pests. I think there is a mechanism to bring them from here to the human because I think that might be the gap.
So as you rightly said, there's a 1 year program, I hope you know very well, now under the AIC-CCMB, National Security Disease Control, and then we are taking all of the veterinary doctors and the other doctors, allopathic doctors, and the remaining the forest climate change. So all the people, the core of the team, and then we have built it up. It is we are working on since last 3 years. We hope in the future we will be strengthened. So it's a very good lecture on the malaria vaccine. As you said, it is very, very costly. You don't want to compromise for the cost, as you said. Efficacy, one of my students asked me a very good question. Efficacy is 20% efficacy. So, to roll out in the government of India, do you think the vaccine is better or Dr. Das said about the phytochemicals? So, phytochemicals are the one the same. It is also working against sporozoites, it decreases sporozoites, and both of them, the mechanism is same. It is in the liver, it's acting. So, what is your vision on this? And then, which is better for India future for malaria elimination?
It's again a chicken and egg story.
So what Dr. Das is mentioned is about treatment. He also briefly spoke about preventive prophylaxis kind of thing. Since like I'm a vaccinologist in the last 20 years, it's preventive health. So we always say prevention is better than cure. So my, from a population standpoint, preventive health is going to give you more richer dividends government program, but at a ground level, the preventive health is not 100% rolled out. There is still space for development, be it phytochemicals, be it existing drugs, because in no country, even if you look at the United States and Europe, where I work for Max, vaccine uptake is not even 30 to 40% at most of the places. So there will exist both preventive as well as a therapeutic approach, and there will be approaches on environmental health as well as chemoprophylaxis and which are not human targeted. So, holistic approach which addresses each of these dots will be important in a country like India. So, the answer whether vaccine is important, drug is important, is impossible. And you, as a policymaker, you will realize very soon if you depend on one only, you will always be facing concerns of supply, concerns of failures, concerns of population impact. So it's always a multi-pronged approach with which
That's
addresses.
rightly you said, sir, because always it's not one, it's a multi-pronged approach is always will be successful in all the government initiatives, all the government programs. And sir, this is for our Dr. Praveen sir. As a CCMB, it's a very good institute, we were doing a lot of research. I personally, I would like to ask you, have you done any of the gene drive mosquitoes? No. So why can't we do some research on the gene drive mosquitoes, where some other countries are successful cultivating the gene drive mosquitoes? What are the challenges? Where can your CCMB can focus on research to youngsters? How you can guide on the research products to come into the treatment or as well as in the diagnosis, or it may be on the prevention side.
Okay. So this is something, it's not that we can't do in India. And I know some ICMR, particularly this NIMR, was trying to do it. Okay. I'm sure India as well as CCMB has the expertise. Okay. And so when I say expert is the concept, right? The training of genetic engineering, molecular and all that. So again, as I said, this would require somebody who is an entomologist with background of collaboration with genetic engineering, right? And rather, it's doable. But I think this will require a dedicated group which should do it. And then as far as the success is concerned, I think the success of genetically engineered mosquitoes is it's promising, but it's not that satisfactory as well. Because there were countries, I remember in some of the South American countries where the genetically engineered mosquitoes were this is put in the field, yeah, and they work at the sterile mosquito. They worked in the first few years, where the cases of because for dengue, the cases came down drastically. After some time, the body came back. Okay. And I think the other approach people I think which maybe more future people are doing are the mosquitoes carrying a kind of bacteria that provides that basically the presence of that bacteria is uncontrollable for development of plasmodium in the inside. And that's what some groups also they are doing. there's a group in NIMR, they'll be also working on it.
Has rightly said, sir. I think all we are sitting from the different paths because the intersectoral coordination is must. Individually or longitudinally, we can't work. Everyone we are working, but we are not coming onto the same platform. So I think, as you said, the entomologist and from the policymaker side and the research program, together we can achieve our goals, I think. So this is for Dr. Manis sir. Can you focus on the, we are developing AI. So how we can introduce our AI methods in our malaria elimination program? Do you have any idea or focus? Yeah. Yes, sir. Yes, sir. Sorry.
So, particularly your experience in my life, we are using two AI-based diagnostics, both for vector and for bioscience. One of them is there is a small project funded by the Australian Indian Council in 2022. We are using that using deep diagnostics. It's called Euro and Golden File. So the thing is, you get a 6 minute picture from the slide quickly with this digital technology. And this developed, we have got two combination out of that. This already developed by Intel, and my own lab is associated with that. Number two, we're working with Japanese students using the device, mobile handheld device for vector cam. So it's asset of 6 mosquitoes. You take any randomly or differential vector, take a picture, preview the recording, then this AI-based repository tells you it's possible.
The diagnosis you are using in the diagnosis.
We are using taxonomical and molecular identification of individual. So while the first part that I said we have already done, and so we can expose them, we will showcase that. Second part will be currently there are individual data because it is comparing with the African writers, it's going to be individual writers. Because let's share any, this head works on this abdomen, the morphology, quite similar to many of our secondary writers in India. Two examples related to
So it's in the diagnosis part and then you're focusing. What I would like to focus on mainly on the surveillance. Because as you know very well, AI-driven vector surveillance. Because we have a lot of tribal area, where the forest area, that the highest areas are there. And as we know, we are going to the elimination. Again, retransmission and recurrence may be possible. So once if we identify any of the AI-driven surveillance, vector surveillance, so that we can do the anti-viral and anti-infect measures. So in this area also we can focus on AI-driven vector surveillance. And in Africa, we can capture mosquito with a mobile scanner also. So these things we can, the younger generation, we can develop in the AI also, we can use in the future. And this is one that we can focus on.
Particular prediction, because most of the data, that data has, Dr. Sahu said, requires the real-time monitoring in the field first. So basically has to run from the previous system. And that will usually help in predicting the next episodes of malaria experiment.
So one of the examples I can give you that I was in the University of Tokyo last year, and one of the data that was presented there, they were using this drones to map the puddles around endemic areas. They are the real-time images, and quickly using this model that is actually predicting that here the bacterial capacity
Exactly. So I think in India also we need that. Now when we are going for the malaria eradication, that is required. Drone-driven AI methods to capture the pictures where the source of infection is there, and so that we can focus on particular thing.
Another thing that I wanted to share with you that while Dr. mentioned that my is leading some of these genetics that you see in mosquitoes. I think there is a group in Madurai, the Center for Research in Medical Entomology, and BCRC, Tamil Nadu. Dr. Abudas is at the moment about there, with the director of this senior RNRC. He was instrumental in designing what we call biosphere model. So literally, with all the ideas of Rameshwaram, this idea was developed. Although that study didn't get funded, but I studied the plans regarding IMR and IC were able to end. So this was in collaboration with the UCSF and from the Australian researchers. I think Australia, as you mentioned, is only rolled out in some parts of North, like America and Australia. So actually have reduced the mosquito population and has screened how much of the transmission is controlled. So I think India also there on our event, it's about the kind of funding and the kind of implementation series.
Patient dependence, it's a good dependence we are doing. So I think to the younger generation, we have to work on the AI-driven anti-larval measures and as well as fighting out the mosquitoes wherever it is there. Sir, at large, I would like to ask you, sir, in this gold mine we are going to get it, as you know very well, multi-sectorial line with the multi factors. We have reached to the, it's a very good, the stages which, like stage 0, almost all the stage 3 API more than 1. And then we reach to the API less than 1. The meaning of the disease, 160 disease, in this last 4 years we have reached. As a researcher, what do you expect the governments to do more research in your field with other intersectoral departments? What type of collaboration you need? What type of research you would like to do it along with your things?
Yeah, the intersectorial engagement is one of the key challenges to be addressed with innovative programs like the or the Mandala project in MP or the other project that Hyderabad show us. One of the key is the amalgamation of data across the department, which is done with IMD. And you need the data, you need yes and no, and it is hard to integrate everything. So
I hope you know about the IHIP. It's a real time capture. We are capturing. Yes. I think, sir, what I'm expecting, because we are different areas we are here. So all the students, not only for the PhD side, allopathy, Ayush, and all together, the treatment we have to focus on, as Dr. Das said. And the second one, as Sir said, about the interdisciplinary coordination, it's a must. Interdepartmental coordination is also a must. As Sir said, for the research and for development of vaccines, maybe because we are supplying to the Africa, but the scope for the malaria vaccine in the northeastern is also much more, as you know very well. Even still, the Tripura and Meghalaya, even still we are suffering with the vaccine, as you know very well. So introduction of malaria vaccine with high efficacy, that is the most important. Because 30 to 50, as you said on your discussion, 30 to 50 efficacy, putting in the program a malaria vaccine, which is really challengeable. Can you make, because it's okay, minus 50 and minus 60, how the government will make the transport and all, how they keep it, that is a challenge to the government of India. But how can you, you as a Bharat Biotech representative, how can you do the, as a programmatic side, how can you do your favor from your biotech, Bharat Biotech?
first of all, 30 to 70% efficacy is not a small efficacy. Every vaccine that you see, they are not 100% efficacious. If you look at typhoid vaccine, it has about a 50 to say
and the number, how it will become. Coming to making a humanitarian vaccine with 100% efficacy, it's government setting, it's not true. Because when you develop vaccine, you do not develop a solution. When we start the program also, we have the government, for example, right now we are working on a TB vaccine. We are not closing the door inside the company and making a vaccine. We go to ICMR, we go to Central TB Division, we go to the existing vaccine procurement policies, the price of the vaccine that we procure today for BCG. We get all these things into consideration before we take a decision for the vaccine. Because we also know we are not taking a vaccine to just with the communication. We make a vaccine to the children. We know what the vaccine is in 10 years from now. What is the value that we are giving today to make a vaccine? So, if we eventually make a vaccine to handle with a 90% consumable vaccine, so certainly reach out to SMR and then institute to explain to them that this is the bottleneck, this is always the bottleneck. So if the government says that I will not buy a vaccine tomorrow with 65, I will take the program. Because we don't take a vaccine to make the application.
Please.
I'm not a background on this, but on the biologist, so this 40 to 70% efficacy is huge. Because think about in Africa, who are the groups that die of malaria? They are children below 5 years of age and pregnant women. So even if that's a 40%, so we are saving 40% of those children who otherwise die of malaria. And this 40%, even this 40% is humongous, because we don't have anything better than that. But I think one thing people realize that vaccine is for only transparent. That has nothing to do with Vivax. So in Indian settings, since we have Vivax and transparent both are different person, even if we start it, so this will go down drastically. And moreover, in India, most of the people who die of malaria, they are not children. Because, yeah, all of them. So I think this vaccine efficacy, and we don't have anything, and when the suffering will be children below 5 years age, in pregnant women, I think that's fantastic.
Thank you. Exactly you said so. And then we'll conclude with just tell me within 90 seconds, we can close it, what we can do, what we should not do in the malaria elimination program. This is for you, Dr. Ravi. In 90 seconds, just to tell me, we are going for the World Malaria, today is the World Malaria Day. We are going for malaria elimination. What we should do, what we should not do.
Difficult question. I mean, I will try to answer. But I mean, with my African experience, that with what we have done, it's a place that you have to take as a multi-pronged approach. Where the school teacher, the Anganwadi workers, the smallest division, the smallest village division, people who do alternate medicine also, everyone has to be brought on the same stage. It's not a competition that you create at the grassroots level, rather it's a facilitation of multiple stakeholders. So the onus lies on government and private players or researchers to bring everyone on the table in the work in the way to say that I will do my best, I will contribute what is expected of me, and to bring in most importantly the entomologists also on board and make an equally part. Because I take from my experience, after 30 seconds more, when I used to work in a primary health center, the veterinarians do not feel comfortable to come and sit on the same table. Because you have made this, or we have made this a society or the government system, that he is minus us. Whereas you go to Western countries, the veterinarians earn much more money than the human clinicians. So the more you enable people to express themselves, and all that will be more fruitful if you empower people and enable them to speak their mind and do their work collectively than they will do this.
You should not do it. I think
I should not isolate people and make them say, you are next level, you are researchers, you do not know medical science. It's something that we have to forget because this is what in the West that practice. Everyone is empowered and be on the same stage. Then saying that you are a researcher, I'm not a cardiologist. Should not happen in terms of this tuberculosis or malaria.
as you know very well, so in the COVID, we all of us together, we worked together. Because there is a genomic sequencing. And the CCMB has come to the government of India, we have recognized. And then we have a committee, so in that committee, CCMB also is a part of it. So this should be continued, as I say. Madam, Dr. Kotisha Ramagaru, and then we will tackle what we shouldn't do, what we should do. We all know.
We should fight, once we should do that, we should fight together. Eliminate them by micro, as a federation and recognition, as a human being, I want to contribute for micro surveillance through my students and also through my known public health and also other sector. So we will unite together, we want to fight to further malaria, eliminating malaria from country. And what I should not do is, as I am because we all these days, malaria is, I am putting a last option. Now, so I should not do that.
Thank you, ma'am. Sir, Dr. Medicine Ramagaru, can you focus what we should do it and then what we have to do?
Covered some parts of it is already favorable. From our research is coming from a diagram of the international cycle vaccines in and in the last 20 years of research, that we can see is first thing what we should do, we should do away with two other discussed. The diagnostic vaccines, the drug vaccines, and the mixed vaccines. All at the level of 2 So, diagnostic is very because I'm going into that particular WHO non-clear based Number two, for example, is alternative medicine. People you will take it off the third, get all the research institutes within their idea of to map or create a dashboard for research inside the so we should do this. What we should do also is develop those intersectorial engagements. Fourth is keep funding those indigenous methods. Mandala project in India, one project in Odisha, and several others, and we not be getting exposure. So all these things at the level of land and at the level of these are what we should not do is the limitation that I think is not having compliance. Compliance with vaccine, compliance with because India is not doing the vaccine, but we are doing the inside. Not and we will see the shortfall in compliance. People do not come forward. This is an example. You go to people in this country, this person knows you, and then you have to say, he said that I am feeling healthy. We talk about asymptomatic malaria. How do you convince him? He is helping. He is cutting boards, he is selecting.
You rightly said, asymptomatic carriers are increasing every day. That's a challenge, it's a big challenge.
It's the participation. How do you do that? Like I said, the word typically is Asha is very good. It is Asha is always overburdened with several months of work. So probably workforce increment. But you have to work in the otherwise if you go and tell me, we should participate more
Exactly, that was yes, absolutely. And then sir, Dr. Purin sir, and then can you just focus on what we should do, what we should not do it
Yeah.
for this summary.
20 seconds.
90 seconds.
So I think we should invest and expand in disease epidemiology and biotechnology. That's what we should do. What we shouldn't do is don't take antimalarials just to have fever. Because that's actually what happened in Africa. And the chloroquine was used like a candy and the drug resistance spread to the level of almost 100%.
It's a nice discussion. Sir, I'm sorry I went first because I was confused with the Dr. Praveen and Dr. Puran. Okay, so Dr. Praveen, thank you so much. I think, thank you. Thank you, Madam, Dr. Kodishwara Magaru, and Sir, Dr. Kadri 2026 has come. So one more year is there. But our